Good morning and welcome to ABG's Life Science Summit. My name is Adam Carlson. I am a equity research analyst covering pharmaceutical and biotech companies. Over the next 2 days, we'll hear from over 50 companies, ranging from smaller biotech and medtech companies to more established pharmaceutical and medtech companies. We're running dual tracks on this channel, primarily biotech companies, and on track 2, primarily medtech and life science tool companies. With the exception of our fireside chats with AstraZeneca, Novo Nordisk, and Sobi, the format is a presentation, company presentation for around 20 minutes, followed by 5 to 10 minutes of Q&A. And I'm pleased to welcome our first company, Hansa Biopharma, and we're joined by Hansa's CEO, Søren Tulstrup. Hansa is a Swedish-listed commercial stage company, market cap around 2.5 billion Swedish krona, leveraging its enzyme modulation platform to enable modulation of autoantibody levels in kidney transplants, initially with its recently launched imlifidase, but also in rare antibody-mediated autoimmune conditions and gene therapies. I'll pass over the word to Søren.
Thank you very much for the introduction. Good morning, everyone, and thank you for your interest in Hansa Biopharma. Before I start my presentation, just the usual warning that the presentation today contains forward-looking statements, and as such, you should apply appropriate caution. I'm sure some of you are not too familiar with Hansa Biopharma, so here's a snapshot overview of the company. The company was founded back in 2007 as a spin-out from Lund University. And what led to the foundation of the company was the identification, the discovery of an enzyme today known as imlifidase that has the unique property that it very fast and effectively cleaves immunoglobulin G. And it was thought that that could have therapeutic relevance. If you then fast forward 15, 16 years, today we're a commercial stage company with our first product on the market in Europe under the brand name Idefirix. That's in kidney transplantation. We've also moved into autoimmune diseases where we've generated what we think is very strong proof of concept in a disease called anti-GBM or Goodpasture's disease. And we've moved into the gene therapy space through partnering with Sarepta Therapeutics and also Bayer. Just looking at Idefirix in Europe, we think that there is very significant commercial potential. This is the first and only product approved to desensitize these so-called highly sensitized patients that have a hard time finding an organ that's a good enough fit for them. And they typically die waiting for an organ transplant rather than be transplanted. So there's a high degree of unmet medical needs. We've also, as I just said, moved into autoimmune diseases with anti-GBM, but there's a host of other diseases in that space, and we have an ongoing phase 2 program in another disease called Guillain-Barré syndrome. And we're also looking at post-transplant situations where there are issues with organ rejection. We have grown quite considerably over the past few years as we've become commercial stage. Today we have approximately 150 employees throughout Europe as well as in the US. We last raised capital back in the summer of 2020, and we're financed well into 2023. So the past couple of years obviously have been quite challenging for humankind in general, given the pandemic and the war in Europe, etc. But it's actually been a pretty successful period for Hansa Biopharma. We have moved forward according to plan with the launch in Europe, getting market access, pricing and reimbursement at the appropriate price point. Most recently, we got the pricing and reimbursement in Germany. We've also gotten early access in France. We've started to partner with companies to look at markets outside of the EU where we can leverage the EU approval, like Israel and Switzerland. We've also partnered looking at our platform activities with a number of different companies. I mentioned Bayer and also Sarepta Therapeutics in the gene therapy space. We've also partnered with Argenx looking at the autoimmune disease space and transplant area. And the organization is certainly a very experienced one, but also a highly motivated one. We've now, for 2 years in a row, been certified as a great place to work based on employee feedback. So just one slide on the platform, and essentially we have a number of different enzymes. They all have the same mode of action. They cleave immunoglobulin G, and they cleave all 4 subtypes, and they're specific to IgG. They do not touch other immunoglobulins. And what happens is that the molecule is essentially decapitated, if you will, just below the so-called hinge section, creating a FAP2 and an FC component, thereby rendering the molecule inactive almost immediately, so that within a couple of hours from a 15-minute infusion, there is essentially no activity. You can see that levels of IgG drop down below detectable levels. And they stay down there for approximately 7 days before gradually bouncing back. And then within a month or so, you're back to normal. So imlifidase is produced by a human pathogen, and it's very efficacious, as you just saw. It's also well tolerated generally. It has one drawback, and that is the fact that since it comes from a human pathogen, it is immunogenic, and therefore we're not developing it for very frequent dosing. But just looking at single dosing indications and infrequent dosing opportunities, there's quite a few of those. Already we're active in the transplant space, as you can see here with the kidney, but in addition to the kidney, there's obviously other organs like lung and heart, where you have similar issues with the sensitization. We're also active in the autoimmune disease space. I mentioned anti-GBM, where we have very strong proof of concept, but we also have an ongoing trial in Guillain-Barré syndrome. These are hyperacute monophasic autoimmune diseases where essentially you only have one attack, and if you can deal with that successfully, the patient is generally well off. So they are kind of right in the sweet spot, but there are other similar types of indications in the broader autoimmune disease area. Then in the gene therapy space, the problem we're solving for is the presence of neutralizing antibodies against the AAV vectors, very frequently used by gene therapy companies to get the healthy gene into the cells. And here we are currently focused on Duchenne muscular dystrophy and limb-girdle muscular dystrophy. And we're also now, through our Bayer partnering, focused on Pompe disease. But there's a very broad range of ongoing programs where you have significant issues with neutralizing antibodies against the AAV vectors used. So there's quite a significant opportunity in the broader space of gene therapy. And then lastly, there's also some oncology indications like hematopoietic stem cell transplantation, where we think imlifidase has potential. Then we're looking at another generation of enzymes that are less immunogenic, just as efficacious as imlifidase. That's the target product profile at least, and well tolerated, but less also immunogenic, so that they can be dosed more frequently. And that obviously opens up to a very broad universe of more chronic diseases, both in the autoimmune disease space and in the transplant space, the gene therapy space with multiple dosing, as well as in oncology. With so many different opportunities, obviously it's very important that you have clear priorities, and we have a very clear business model in place. This is what you see depicted on this slide. At the core of our business model, we have our enzymes, and they all have the same mode of action, as I said. Then as we develop these into drug candidates and approved products, we aim to essentially control most of the elements, the key elements in the value chain towards the market. Doesn't mean that we need to own them, but certainly we need to control them so that we get the majority of the upside, and also so that we can ensure successful launches. But then as we approach the launch time point, depending on whether it's a product in a space where we have most of the knowledge needed to succeed, and where it's generally relatively easy to operate because it's a concentrated target audience, etc. If that is the case, we will by default try to launch ourselves in most of the countries. If that's not the case, then we will partner. If we need access to complementary assets and so on, for instance, in the gene therapy space or in the oncology space, we will seek to partner to have our partner essentially really help with the launch, or maybe take care of the launch altogether, and in some cases also late stage development. Let me say a few words about our lead asset, Idefirix, for kidney transplantation. As I said, there is a very significant commercial opportunity, we think, in Europe first and in other geographies later. Looking at the European Union, where we have approval currently, the label we have is desensitization therapy of highly sensitized patients that are unlikely to be transplanted and who have an organ from a deceased donor. That is actually a pretty significant opportunity, even though it is, of course, restricted. It is quite significant. Generally, it's agreed that if you have what is called a CPRA rate of more than 80%, you're highly sensitized. That's 10 to 15% of all patients on the wait list, and quite a significant part of them are unlikely to be transplanted. And the majority of transplants taking place today are from deceased donors. So just looking at the European landscape and here also the US landscape, there are approximately between the two countries, you have a little bit less than 200,000 patients on the wait list. Given the fact that there's an undersupply of organs, it's only 1 in 4, 1 in 5 that are actually transplanted. Over the past few years, the number has grown. Last year, or in 2020, which is the last year in this slide, there was an impact from the COVID-19 pandemic, but in general, it's been growing over the past few years. And so just looking at that, you're looking at how many patients that are on the wait list, you take into consideration the fact that there's an undersupply of organs, you have an addressable patient population in each of the two major geographies, Europe and the US, of approximately 3,000 to 4,000, so altogether 6,000 to 8,000. And we will get a proportion of these patients. But we won't get them from day one. When you launch a very innovative product, you really need to put the infrastructure in place, change the mindset, etc. So initially what we're doing, we're really focused on the leading transplant centers. We are making them clinically ready, commercially ready, and then we want them to use the product in the right patients and have good first experiences. Typically, they will wait a number of months, having treated the first patient, before they decide to continue and use it on a second and consecutive patients. So that's why what we're looking at really is an S-shaped launch curve, where in the second phase, when we're moving into more exponential growth, you're getting repeat business in the centers, and you're also getting new countries providing market access by giving price and reimbursement. And in the later years, we will also aim to broaden the label and so on. So that's why you see some growth also in the later stages. This is a map of Europe, just to give a snapshot overview of where we stand in terms of market access. As I said, initially, we have now gotten access in quite a number of countries, Finland, Sweden, Netherlands, most recently Germany. We're also accessing the French market through an early access program. We're in discussions with NICE in the UK and with the authorities in Spain and Italy and so on. And we expect that by the end of this year, we will have access to the majority of the countries in Europe. In the US, we have agreed with the FDA to run a pivotal phase 3 trial. It's going to be, or it is ongoing, so I can say it is a relatively small trial in 64 patients, where they will be randomized when they get an organ offer. They're highly sensitized. They get an organ offer. They will then, and if not a good enough fit, they will be randomized to either desensitization therapy using imlifidase in the active arm, or in the comparator arm, standard of care, which for the majority of these patients will be to just wait through the study period, which is 12 months. Hoping against hope that an organ that's a good enough fit will be made available so that they can be transplanted. And we're using EGFR as a surrogate marker of efficacy. If the patients are not transplanted, they've allocated a value of 0. So it's highly powered to show benefit in the active arm, even though it's a small trial. We're currently enrolling patients here. We aim to have it fully enrolled by the end of the year and have high-level data readout by the end of next year, so that we can submit a BLA under the accelerated approval pathway in the US in the first half of 2024. Snapshot overview of our pipeline. It is a rich pipeline already, even though we're a young company. Idefirix already approved in Europe, moving forward with a pivotal trial in the US, and then there are other geographies as well we're looking at. Then we have ongoing trials in AMR, which is a post-transplant indication, Guillain-Barré syndrome, anti-GBM. Here we're starting a pivotal trial. We have agreed with the FDA and the EMA to run a pivotal trial in 50 patients. And then in the gene therapy space, it's still preclinical work with Sarepta as well as with Bayer. With Sarepta, the work has progressed quite a lot, and we hope that the animal studies will be concluded this year, and then it's up to Sarepta to take a decision how to move this forward. And then the next generation enzymes, the so-called NICE program. Here we're doing IND-enabling tox studies right now, and we aim to have these completed by this year. So I think I've talked to this already. We have a platform that's quite versatile and flexible, can be used in multiple broad indication universes. Right at the center, we have these enzymes. We can also combine them with other products that also target IgG. We can use them hopefully both in an acute setting as well as in a more chronic setting. And we're active in autoimmune diseases, transplantation, oncology, and gene therapy. Just one slide to underline that, as I said initially, we really have generated a very strong proof of concept for the whole platform in the autoimmune disease space, looking at IgG driven autoimmune diseases such as anti-GBM. And here is just the high level readout, if you will, of the results from this phase 2 trial with 15 patients. Normally, 2 out of 3 patients end up in dialysis. When we started this trial, 2 out of 3 of the patients in the trial were already on dialysis. Using then imlifidase, we were able to prevent all of the patients that hadn't progressed to dialysis from progressing, and even some of the patients that were already on dialysis were able to be taken off of dialysis. So that's really very, very strong data, albeit in a limited number of patients. The AMR trial is ongoing right now, just like the Guillain-Barré syndrome trial, phase 2 trial. We're looking to enroll 30 patients. We have 29 at this point, so it should be fully enrolled very soon. And then we should have data, high-level data, in the second half of this year, which we're certainly looking very much forward to. Other milestones looking forward is Guillain-Barré syndrome. Also, we should have that fully enrolled this year, and a readout then 6 months after. Looking, to 23, we should have data from the ConfIdeS trial, the US pivotal trial in kidney transplant, and a BLA submission in the US in the first half of 24. We've not put the gene therapy milestones here because it's still too uncertain, but we also expect hopefully very exciting news in that space over the coming years. So we have had a couple of very interesting and successful years behind us now, and now we're looking into an equally exciting future. And that's all. I'll hand back over to you, moderator.
Perfect. Thank you very much for that presentation. A few questions then. First one, you referenced your European label, deceased donors, highly sensitized, typically cutoff being above 80 on the CPRA. And I'm just curious, with the pricing reimbursement discussions you're having, how, and I know you can't give precise details, but how broadly aligned is the label to the populations that you're being reimbursed for? Are you quite happy that you're getting a similar sort of addressable patient population as it relates to the CPRA score or other parameters?
Overall, yes, we're quite happy with the outcome of our discussions with the payers in the different European countries. Obviously, they differ, right? There will be different perspectives and so on. But in general, certainly it's within label, right? So it's not a new population. It's a patient set that we think will have good results from using imlifidase. And it certainly gives enough room, if you will, so that you can actually maneuver and make sure that the product has an uptake in the different countries. There will always be restrictions, and that will vary from country to country. But in general, I would say we're quite happy with where we stand.
Good. And deceased donors is on the label now. And just curious to hear plans you're thinking around living donor donations. The AMR study, as I understand, is in fact at least partly, right? Including living donor donations as well. So you're generating some data there. What would the kind of process be for expanding the label into that subset of transplantations?
So right now, initially, we've chosen specifically to focus on the patients with the highest degree of unmet medical need, and that is patients with an organ from a deceased donor, because today you don't have any remedies that will essentially enable them to accept an organ that's not a good enough fit. You don't have enough time to treat them. But certainly we also see a potential in the living donor space. If you have a living donor and that person is not a good fit with you, it would be great if you could desensitize the recipient so that the transplant could take place. That will also increase the number of organs being put into the pool, if you will, and the number of transplants taking place. So clearly that's something we are looking at, not in the immediate short term, but certainly over time we expect that something will be, this will be a real market opportunity.
Okay. And we've gotten news of positive reimbursement decisions in Germany and France in recent times. And on the Q1 call, you're quite upbeat on the progress overall in Germany. Not perhaps sales necessarily, but the progress you're making there, and just considering the sort of the lead time between a favorable reimbursement and until clinics are up and running, and you might see that on the P&L. How much, if anything, should we sort of be expecting from these particular Germany and France recently approved markets in Q1?
Well, I would say, now we're in Q2. I would say we certainly expect sales from both countries this year, but it's impossible to predict what quarter you'll have sales, and you'll have very volatile sales because the value of one patient is very high. It's 3 million Swedish kronor, 300,000 euros, and the number of patients is low, and it's a one-off therapy. So you'll see this kind of sales. Typically, there's clearly a lead time from getting market access, from getting pricing and reimbursement until you actually see the first patient treated. So it will not come immediately, but hopefully, certainly, we expect it this year. Yes.
Okay, good. And I think you've referenced it as well, if I remember correctly, in the Q1 call, that initial draft guidance from NICE, not recommending reimbursement, not unusual. As I understand in terms of how that process often goes. But you seemed, upbeat as well on the potential to secure reimbursement in the UK. Then, compared to then, has anything changed? Are you still hopeful for reimbursement in the UK?
We're absolutely still hopeful. It's always a tough battle to negotiate with NICE, as everyone knows. But we think we have a very good case. We have strong local support. Most recently, actually, we were nominated as one of 3 products for best pharmaceutical product in the UK in the Prix Galien, the UK award ceremony. So there's clearly focus on this product, the opportunities and the benefits, and so we think that in the end, NICE will say yes, and we'll be able to agree. But it's always a process that you have to go through, and sometimes it takes. Not that long, and sometimes it takes longer.
Yep. And is there any thinking in terms of clinical trial sites experience, clinical experience in the UK, perhaps less than in some of the other key markets, if I recall, perhaps not. Does that feed into it at all? That the UK is sort of less accustomed to imlifidase in terms of that negotiating process being more drawn out?
I don't think so. I mean, it's a very tight-knit international community, and we have very good supporters in the UK. It's just a question of different processes and different approaches from the authorities, and so as I said, I'm optimistic that we'll be able to get market access in the UK as well.
Good. And in terms of funding needs, you're funded well into 2023. I believe you said in the Q1 call, and you've also had this message of sort of exploring various options for financing, presumably suggesting something other than equity. And I'm wondering if there's anything more you can say there in terms of what are those options. Presumably, debt financing would be one of them. Are things like selling royalty rights or things like that, more atypical things, seriously being considered as well, or how should we be thinking about that?
I mean, I think all biotech companies are operating in these choppy waters right now in the biotech sector. I mean, you're forced to look at several different options, right? And we're certainly doing that. We're in a very good shape given the progress we have in our business, given the fact that our platform is validated, given we have a product on the market, etc. We're a mature company, so it's just a question of finding the optimal solution in this situation. And obviously, you're looking at apart from equity, you're looking at structured financing. You're looking at other sources of non-dilutive capital. We're very focused on not diluting our assets at this point in time. We think we're best placed to develop them and get the benefit out of them.
Okay, good. Maybe just one last question. You've spoken about guideline work, the ESOT guidelines, and getting imlifidase in there. The progress on that side of things, and how material do you think that would be? I mean, as you're saying, it's a small, tight-knit community and so on, and presumably people are sort of well aware of days. How big of a difference is it being formally included in that guideline, would you say?
It is certainly helpful. Initially, what is critically important is that you have local protocols in the specific centers, right? So we're working very closely with the leading centers across Europe, and that is happening as we speak. But in addition to that, it's certainly supported also to have European-wide guidelines. And here we're working with the European Society of Organ Transplantation. There's a specific task force in place looking at updating guidelines, putting guidelines in place specifically for desensitization treatment of highly sensitized patients, because that really hasn't taken place in Europe so far. And that's going to be helpful, absolutely. Short term, you will not see any immediate impact, but over the next years, absolutely. This is really helpful, and I'm very encouraged by the support we're seeing and the seriousness this is approached with.
Perfect. I think we're at time. Søren, thank you very much for an interesting presentation.
Thanks so much for inviting me. Thank you.
The next company to present is Cinclus Pharma, and we're joined by their CEO, Christer Ahlberg. Cinclus is a Swedish, currently unlisted clinical stage biopharma company developing small molecules for gastric acid-related diseases. It's got a lead asset, linaprazan, in a phase 2 study for GERD currently. Linaprazan may be a familiar name to some as a former AstraZeneca drug candidate, and Cinclus also a familiar name to some as a link portfolio company. I'll hand over the word to CEO Christer.
Okay, thank you very much. Thank you for the introduction. We can go on to the next slides, please. And next again. As you mentioned, I'm Christer Ahlberg and I'm the CEO of the company. And today's presentation will be about Cinclus Pharma. I will guide you through, first a short introduction of the company and also our therapeutic area where we are focusing on, and also our lead candidate, linaprazan glurate, and the development steps for the future and the strategy for the future. So that will be the next 20 minutes. So please go ahead to next slide. And next. Short presentation of myself. I have been within the company since last summer, more than 25 years within the pharmaceutical industry. Recently I came from Sedana Medical, which I took to the, which I made an IPO for 5 years ago, and also got the approval of their therapy. I also worked as a CEO for Unimedic, one of the biggest company within the MedCap group. Also established Eisai, a Japanese company here in the Nordic and Baltic as CEO. Also during the time when Eisai and BioArctic had initiated their collaboration. I was also part as a marketing head of MEDA in the beginning of the MEDA journey. I was headhunted by Anders Lönneden from AstraZeneca before where I started my career. I also participated in the launch of Nexium. So that's my short history. Going to the next slide. So looking into Cinclus Pharma, as we already heard from the beginning, we have one asset, very interesting asset in a drug candidate called linaprazan glurate, which is addressing a large unmet medical need within the gastric acid-related diseases. Market potential is huge, of $5 to $10 billion annually for our targeted segment. Our candidate do have a very interesting profile with the superiority efficacy potential and interesting pharmacokinetic profile. The ongoing phase 2 study is coming to an end, and we are estimating to have a top-line result of the summer here. And the clinical program by itself is also de-risked quite a lot, I would say. Most likely, we have the active metabolite linaprazan has been tested in more than 2,500 subjects by AstraZeneca, and also within a lot of animal studies before. We have an excellent biomarker which actually predict the clinical outcome later on. I will show you soon. We have a good patent situation, 34, and with the additional patents valid submission, and if they come through, then we will have approval until 4041. Huge experience within this field in the company. Both CSO and CMO have been part of the huge successes of Astra and the development of Losec, Nexium, and linaprazan. So definitely we have a potential to become best in class and a future global market leader here. So next slide, please. I will also, before going into any other details, I will give you just an understanding of this market, so you understand the different, the importance, what we need to do here. Gastric acid-related diseases have always been a very attractive market for many decades, but each paradigm shift has been followed by that you increase and improve the acid control in the stomach, where you how long time can you stay above pH 4 in the content in the stomach. And the better control you have, the better chances you have to get a market leader position. That was the reason why PPIs became so popular, and also Losec became the biggest drug in the world, as well as the follower Nexium also became the biggest drug in the world. And that is also one of the reasons why the new generation and the new class PCABs, which is now launched in Asia and especially in Japan by Takeda, where you're also improving the acid control even better than PPIs. And now you can already see that Takecab, which is the Takeda's PCAB in Japan, has the market leader position, and actually they have selling for $1 billion only in Japan, and is now the biggest drug in Japan all over the market. So that's enormous, actually, I would say, showing the potential. And our PCAB, which is representing the same class, a new class, actually have the potential to show even better acid control. So definitely it's a very interesting product. So next slide, please. But before going into details, I just want to give you some updates of the disease. Next slide. This is a not very complicated disease, but patients suffering a lot due to erosive gastroesophageal reflux disease. And the intention with the system here is that it should be one-way direction. All the content in the stomach should go in one direction, not going upwards. And that is the problem when the ring muscle between the stomach and the esophagus doesn't work as it should. When stomach content goes up in the esophagus, and that gives very severe symptoms sometimes, but also erosions in the esophagus, which is not good. There are different alternatives to treat this. You can do it with surgery, not very popular these days. But this was something that when PPIs came on board, they succeeded actually to reduce the pH or increase the pH level in the stomach to above 4. Which is important, because if you are above 4, you don't get any erosions. But the problem with the PPIs, they couldn't do it for 24 hours. Approximately 12 hours was but they were very effective in 12 hours there, unfortunately. But so what we have seen is that approximately 30% of the patients are not fully sufficient treated with the PPIs currently. So there is a huge group with the unmet medical need of 30% of the current treatment group. Next slide, please. So our focus will be the erosive patients suffering from this disease. And that will be the initial focus for our indication ambition as well. Next slide, please. So looking in the indication aspiration we have is to go for the full grade, full e-GERD indication, all different grades from A to D, different severeness of the disease, both the healing phase, but also the maintenance to prevent relapse and also relief of symptoms. So that will be the indication, but our focus will definitely be on the 30% that is not sufficient treated by PPIs and the most severe e-GERD, and patients that actually need to improve the quality of life and symptom relief. Next slide, please. Looking into the market potential here, I mentioned 5 to 10 billion US dollar, and that is calculated on this epidemiologic data here. If you look into North America and Europe only, this is a huge very common disease. 175 million people are suffering from this, but and 40% of them are suffering from the erosive disease then. So that is the indication, the 70 million people in Europe and North America. But the focus target group will be the most severe, C and D grades, and patients that are not sufficient treated with PPI. So we're talking here about approximately 18 million patients. And with an average daily treatment over a year of 100 days, and a price of $7 per day in US and approximately $2 in Europe, gives you market potential of $7 billion US dollar. And then, of course, you have an upside of in the rest of the world, which is this is also, I mean, this is commonly used also everywhere in the world, but that's definitely an upside. So going to next slide. Just going through the substance somewhat, and next slide again. I mean, I don't want to go too much into the details here, but this substance is developed by Astra, and especially the active metabolite linaprazan was developed by AstraZeneca. The linaprazan was a fantastic, effective drug. They had, but after, and they actually, the intention was that this should be the successor to Nexium. They had obviously two problems with linaprazan, which they faced after the phase 2 studies. Short duration and a peak Cmax, which really peaked initially after the first dose. They tried to compensate the short duration by higher doses, but since you had this huge peak in the Cmax, also gave them some challenges when it comes to side effects. So our founders, Kjell Andersson and Mikael Dahlström, the inventor of linaprazan, they was employed by Astra during that time and was part of the Astra journey for many decades. They started to find different solutions on these two issues, and they actually found a solution, and they made a prodrug, which actually prolonged the duration significantly and decreased the Cmax, the peak, by 75%. Which means that you actually mission was completed. So now we had a substance that actually was had a long duration and full acid control without safety or side effect issues. But the problem was that Astra, in parallel to this, had decided on highest level to actually stop working with the GI business area. So they decided to not continue with the GI business at all. And during that time, it was a possibility to have some kind of management buyout of the substance, and we could actually establish and they founded Cinclus Pharma. So going to the next slide. And the biggest difference between a potassium competitive acid blocker, PCAB, which is our class then, the linaprazan glurate class, compared to PPI, Losec and Nexium class, is the main difference. I will not go through. It works approximately the same, but there are some main differences clinically. As I mentioned initially, is that PPIs only have a half-life of 1 to 1.5 hours, which is way too short to be effective over 24 hours, even though you take three doses per day. And the problem also is a very slow effect onset, and takes 3 to 5 days to get full effect. And this should be in comparison to a PCAB, where you have 10 hours with in half time, which give you full 24 hours control, and you have a very fast onset. You have full effect within 1 to 2 hours. Huge difference. Going to the next slide then. I also mentioned about the excellent biomarker. This is also important to understand to predict the future studies. This curve is the result of decades of science and a lot of studies, and it shows the correlation. If you can keep the pH 4 above 4 over 24 hours, all patients will be treated and healed from the erosions. That is a very, very strong correlation here. So you know, if you can fix 24 hours acid control, then you heal the patient. So going to the next slide, looking into how the drugs are succeeding with this. Looking into the left paragraph is Nexium. Here you can see the green curve is the first dose, showing very slow effect. That you see the time after dose up to 24 hours, and then up to the pH 4, and you see the dotted line. That's the borderline of pH 4, which you need to come above to make sure that you heal the patients. And the first dose is barely coming up above pH 4, very slow onset. After 5 days, you are quite good in that, but still during evening and night times, you have problem to achieve pH 4, above pH 4. So many hours without acid control. Looking into Tachycab, our competitor in the new first PCAB came to the market. You have a similar curve, actually. The difference here is that you have already after first day, first 2 hours, acid control about pH 4, but you're dropping again late evening and early night. This is not optimal for the most severe patients. Going to the next slide. What we can see here is the comparison when what we have done in our studies. We can see that you have a fantastic curve when you have the right dose. You have full control in the yellow curve that obviously give you 24 hours acid control. No, you don't drop below. This is our drug. This is linaprazan glurate, and this is interesting to see. If you compare that to the next slide, between the different dose, of course, this will have a clinical effect differences, and this is what we are now looking into in the phase 3 studies to show. And looking into the next slide, we are now finalizing the phase 2 study. And what we have seen is that this is a dose-finding study, and of course we have a comparator in the study in lansoprazole, but it's power to show the differences in doses to be right when we're starting up the phase 3 study early next year. So going into the summary of this, next slide. We have a de-risked development program where we have a good relationship with the authorities, both in US and Europe. We know what they are requiring, and we actually are working on to deliver that, and we are confident that we can do that. We have the linaprazan, which is the active metabolite in our drug, and that has been studied in more than 2,500 patients and in all the animal studies, and no safety issues there, which we are also now confirming in our tox studies. And we have these active metabolites, which I have shown, which will definitely predict the outcome in the future clinical studies. So with that said, it looks very promising. Looking into the commercial factors, next slide. We have already said what is very much important to show acid control, but also looking into the next slide, I think it's interesting to see, next slide please, to see the performance Takeda has done in Japan, where they are now market leader. And this is a proof of concept that PCABs, firstly, there is a medical need out there. PPIs didn't do the job fully. So, of course, we need new drugs. And also PCABs is the new class that actually can meet this medical need. And going to the next slide, I think, though, it's important to differentiate versus PPIs. we definitely have a differentiator factor commercially with a shorter, with a much faster onset, hours instead of days, but also with better superior effect, which is important. Versus the other PCABs, we can say that we have, thanks to the cut in the Cmax, where we are now, we can use the full potential of the substance and have a full dose, and that gives you the full control of the acid during the 24 hours. And that is not the case for Takeda's Takecab, where they are. We believe that they have same issues as Astra had initially. So we also think that we can meet other symptoms that they will not fulfill. Okay, we can. I see this time is running. We can go to the next slide. I just wanted to mention we will not be alone in the market. We are estimating to have a full approval in 2025. Vonoprazan Takecab will be approved a little bit earlier than 2 years before us in the US in this indication. But as we have seen in the PPI market, this market is so big, so there is space definitely for more than one player. In PPI market, we have seen 6 players, and they have all been successful. Here we are estimating to have approximately 2 to 4 players in the western part of the world. There are some more Asian products that do have ambition also here, as you see. So next slide. I can just say the patent situation is good. Yes.
Sorry, just in the interest of time, I think we have to crack on to the Q&A, but we can leave this slide up.
Yeah, we can go to the so we have that in front. Please, next one. There, we can have that.
Okay, great. No, thank you for an interesting presentation. My first question was going to be on how Takeda has positioned Takecab in Japan, and how they sort of regulatory approval and the commercial positioning, as I understand it, being non-inferior to PPIs, but with a faster onset of action. And I'm just curious to hear how that differs from how you're thinking about it.
Yeah, I mean, of course, we will have, if you differentiate it versus PPI, of course, faster onset is important. But superior effect is also important. And since they have only done non-inferior study in Asia, they have problem to do that. So we will have definitely another factor that they don't have, at least not in the eastern part, or not in Asia.
Okay, alrighty.
And also they have focused on independence of food intake as well. You can take this whenever. PPIs you need to take, you cannot take it together with the food.
Okay. And just help us understand, Fathom licensed the US rights from Takeda. And just help us understand why Takeda wouldn't have pursued that US opportunity themselves, or indeed why there was such a lag between launching in Japan and them seeing the opportunity outside of Japan. Why did that?
Well, you have to ask them, but no, I mean, of course, we can always have guesses here, and of course we have investigated quite a lot why. But the problem, I think, from initially, they were a little bit unsecure if the PCABs should work in the western part. Normally, you get a very good result in Asia. You have seen that in the past with all these kind of PPI investigations. But I would say now that you have de-risked the case quite dramatically when Fathom has done their phase 3 studies and actually showing good results compared to what you have seen in Asia. And definitely, I think Takeda didn't maybe estimated that, and it was some kind of de-risk position they had. They will, of course, earn quite a lot in royalty income from Fathom, and I really I think they were a little bit unsecure how this should be managed in the West, but obviously we see now that it works.
Okay. And then maybe just one question on the profile. You were saying how the Cmax has been brought down, and you prolonged that half-life to get that favorable product profile. I'm just curious, mechanistically, is there any reason to think that higher Cmax that Takecab has, for instance, is necessary or a favorable feature in terms of getting that efficacy, and that you need a degree of high Cmax?
No. That is totally useless. Actually,
Okay.
they have. I mean, linaprazan initially they had 20 times higher than was needed to reach pH 4. Actually, we have, as it looks now in our early stage studies, we have even faster onset. So you don't lose anything by cutting it. It's only good. It's good.
Okay, maybe just one final question then. On your IP situation, we didn't have time to look at it. What does that look like? And then separately, there is, as I understand this 5 years of extended market exclusivity in the US if you go after H. pylori as your lead indication or initial indication. Is that on the table for you? Is that something you're looking at?
Yeah, definitely. We are looking into that, and we will do that, and we are working on that now. So, I mean, patent is one thing. We have the current patent is valid until, including the extension, is valid until 34-ish, 33 and 35. But also when you go with a new substance, you get the data exclusivity. In Europe, you get 10 years from registration, so that is at least to 35, and that is as strong as the patent, I would say. In US, you only get 5 years initially, but now with the possibility of a QIDP to prolong, to make H. pylori as the indication, then and as a first indication, then you can prolong it by 5 years. You also get 10 years in US, and that's we will do this H. pylori and eGERD studies in parallel as phase 3.
Okay, Christer, perfect. We're all out of time. Thank you very much for an interesting presentation.
Thank you. Bye.
Hi, and welcome back. My name is Gonzalo Artiach, and I'm a healthcare equity analyst here at ABG. Our next company is Nycode, a Norwegian biotech company developing DNA vaccines for treating cancer and infectious diseases. We have here today with us Agnete Fredriksen, Chief Innovation and Strategy Officer, as well as Siri Turnhaug, CMO. Please, the floor is yours.
Thank you, and thank you for inviting us to this event. We are happy to present the data recently generated by Nycode today. So if you go to the next slide. So I assume you're all familiar with the forward-looking statement. So on that note, we can go to the next slide. So I'm happy to have Siri with me here today as the CMO with many years of prior experience within clinical development and then pharma and medical affairs. Together with me, I'm co-founder and Chief Innovation and Strategy Officer on Nycode. You can move to the next. Next slide. Yeah, so briefly an overview of Nycode Therapeutics. Our vision is to build a leading immunotherapy company. We have so far based all our clinical products on our proprietary Vaccibody immunotherapy platform that uniquely targets antigen-presenting cells, which we have seen induces a strong, particularly CD8 killer T cell response. It is thus based on an adaptable platform that can quickly be used to target new diseases. We have a pipeline currently consisting of products both within oncology and infectious diseases, both partner programs as well as wholly owned clinical candidates. And we are in parallel working with a dual focus on the further potential of a differentiated molecular platform and clinical projects. We have, over the last 1.5 years, been very busy on the business development side and signed 2 out-licensing deals with top-tier partners, which gives us potentially more than $1.6 billion US dollars in upfront and milestones plus royalties. We have a clinically validated technology, including our recent positive interim results from the phase 2 HPV-16 positive cervical cancer program that Siri will go through in detail today. And we are well capitalized with multiple significant catalysts in the near to medium term. We can go to the next. So a quick look at the pipeline, due to the recent release of the interim data, I would like to focus the attention on VB10.16 as of today, where we have recently announced interim clinical data, and we will be able then to announce further updated clinical data first half next year. And we also recently announced that we are moving into HPV-16 positive cancer and head and neck cancer with VB10.16, in addition to our internal programs that we are moving forward, as well as the partner programs. Next slide. Next slide. So briefly look at the Vaccibody technology that we're working on within Nycode. This is a fully flexible modular vaccine technology that consists of these 3 different modules, and where we combine genes with certain properties into one molecule, which means we'll have a fusion protein that has a targeting unit in orange that can attract and bind to antigen-presenting cells, a dimerization unit in the middle that is then connected to the antigen of choice, which can be globular antigens or a set of T cell epitopes from different sources for vaccines developed towards cancer, viruses, bacteria, parasites, and as you'll see today, also autoimmune disorders. Next slide, please. So for the Vaccibody technology as such, our vaccines in the clinic are focused on a naked DNA plasmid-based vaccine that is encoding these Vaccibody molecules. The DNA plasmid will be delivered intramuscularly, and this will lead to the Vaccibody fusion protein being secreted out to the transfected cells. We have included a chemokine in the clinical products as a targeting unit. So it actually then, through chemotactic gradient, will attract antigen-presenting cells to the injection site And the chemokine in the orange part of the molecule will actually bind to chemokine receptors on these antigen-presenting cells, which provides the necessary signals in order to induce an immune response, which includes also that the Vaccibody protein is internalized within the antigen-presenting cells through the receptor internalization, and it's channeled both through the classical as well as, importantly, the cross-presentation pathway, which is explaining why we see a very broad and strong CD8 killer T cell response with our technology. And these T cells then will come and go and recognize cancer cells or virally infected cells and be able to recognize them and kill these cells. So we can go to the next slide. And then I will hand over to Siri that will go through the clinical programs in more detail. First, VB10-NEO, Siri.
Yes, thank you. So yeah, Nykode has 3 clinical development programs, and the first one is VB10-NEO. So let's move to the next slide, please. So VB10-NEO is our fully individualized neoantigen cancer vaccine. We have previously presented that we have shown strong ability to create strong and targeted immune responses within this program. And these responses has also been correlated to clinical responses, as illustrated on the right side of this slide. You can see 2 examples of clinical responses. Initial on the top graph, you can see initially a stabilization of the disease before moving into vaccination, and then a clear deepening and increased response in the patients. On the one graph underneath, you can see a progression before moving into vaccination, and after initiating vaccination, a stabilization of the tumors, which illustrates this what we will show now also for what we've seen with the VB1016 program. So I think you can move to next slide, and then to the next slide again. So VB1016 is our therapeutic HPV directed vaccine against HPV-16. It's an off-the-shelf therapeutic DNA vaccine where we have already finalized one trial we call the VBC-01, which was a phase 1/2 trial run in precancerous cervical lesions. And we are now currently running VBC-02, which is in advanced cervical cancer, and where we recently, as already mentioned, have released interim data that I will come back to and present in some more detail. And then we are planning to expand our program with VB1016 into HPV-16 positive head and neck squamous cell carcinomas within the end of the year. So next slide. So the CO2 trial, as mentioned, is run in advanced cervical cancer, and this is to illustrate the population and the severity of the disease for the patients that have gone into this trial. More than 70% of the patients have received more than 2 or more prior systemic treatment lines. As you can see from the metastatus, several of them have lung and or liver disease before moving into the trial, and 90% of the patients have extrapelvic metastases present within timing of enrollment. I would also like just to highlight the PD-L1 status at baseline, where you can see that 12 out of the 39 patients reported in this interim analysis, so 31% of these patients were PD-L1 negative at enrollment. So next slide, please. Here you can see in the waterfall plot how the patient in this interim analysis are doing. And I think I would really like to highlight the stable disease population, where you can see that 9 out of 17 patients that are stable really show what we call a stable disease plus, so actually a reduction in the target lesions, but not fully reaching the minus 30% needed to become a partial response. So next slide. In this spider plot, it's illustrated how the patients are actually doing. So the green bars illustrates the patients that are in a responsive state, so either complete or partial response. And they are marked, illustrating that we have 8 patients that are either complete or partial responders. 3 out of these patients have fully gone through the 48 weeks of treatment and have still an ongoing response at the time of finalization of the treatment and into follow-up. And an additional 3 patients are still ongoing with response within the study. That means 6 out of the 8 responding patients reported in this interim analysis has an ongoing response. In addition to this, we have several patients, as already mentioned, that are in a stable disease setting, and illustrated here, several of them with really long, durable responses, and several patients have fulfilled the 48 weeks of treatment, and several are still ongoing with responses. So next slide, please. Here you can see some data on differentiation between PD-L1 positive and PD-L1 negative patients. So as mentioned, we had in this population where we had PD-L1 status, we have 12 negative patients. Out of these, 17% have a clinical response, and we see a disease control rate of 58%, really illustrating that VB10.16 in combination with atezolizumab may enhance the clinical response also in the PD-L1 negative patients, where checkpoint inhibitors at the moment as monotherapy treatment are not approved. So next slide, please. So in conclusion from this interim analysis just reported, we see durable responses with a very high disease control rate of 64% in heavily pre-treated advanced cervical cancer patients. We see an anti-tumor efficacy observed both in PD-L1 positive and negative patients with a 27% overall response rate and a 77% disease control rate in the PD-L1 positive patients, as well as 17% response rate and 58% disease control rate in the PD-L1 negative patients. In addition to this, we see a disease control rate of 71% in patients with non-inflamed tumors, including both immune desert and T-cell excluded tumors. We have also observed specific interferon gamma T-cell responses associated with clinical efficacy, and we've seen complete clearance of HPV-16 ctDNA that's significantly correlated with clinical outcomes. In addition to this, from a safety perspective, VB10.16 in combination with atezolizumab seems to be very well tolerated and has a safety profile that would be comparable to checkpoint inhibitor monotherapy. Next slide, please. Next slide. So our last program is the VB10.CoV2 program. SARS-CoV-2 is still a threat and a global health threat. We see still variants of concern emerging, and it is unclear if future variants of concern might be more severe or not. More and more data now illustrates that T cells appears to be central in maintaining protection against severe disease and death over time. In addition to this, approved vaccines seems to have short-lived vaccine immunity, especially visible in vulnerable target populations like elderly and immunocompromised patients. generally, so COVID-19 vaccines induce a broader T-cell immunity across populations, might therefore be a good way to prevent severe disease with new variants of concern moving in. Next slide, please. So the ongoing trial that we are running is a phase 1/2 trial where we are looking at 2 different candidates as a booster for previously vaccinated subjects. So all patients or subjects, in this trial should not have had previous COVID infection, but be previously vaccinated with mRNA vaccines. So we have 2 different candidates. So candidate 1 is an RBD candidate targeting the spike area, similar to approved vaccines, where we are currently still including patients. And then the last candidate, candidate 2, is a solely T-cell based candidate targeting a broader part of the virus outside of the spike area, as an opportunity to give a broader T-cell immunity. And this is a fully enrolled dose escalation trial, and we are expecting data during second half of this year. Next slide, please. Then I will hand back to you, Agnete.
Yes, thanks. Then we will briefly look into the work we're doing on the further platform potential. So next slide. When we changed the name from Vaccibody to Nykode half a year ago, this is because of the further platform potential that we see with our unique know-how and technology. We have currently focused on APC-specific antigen delivery and stimulation within cancer and infectious diseases, as you heard today. We are now doing very interesting work for vaccines against autoimmunity and allergy, where we can take this one step further and look at APC-specific antigen delivery and inhibition. We are also expanding the use of the modularity that we have already worked on within the Vaccibody technology, building in additional modules, novel combination structures, as well as novel therapeutics beyond vaccines. So next slide. So for the 4th module, we have recently released some data where we have been working on how to even further optimize the platform technology. So we call it the 4th module because in the same DNA plasmid-based vaccine, we have been able to incorporate a 4th module that will encode a separate protein secreted from the same cells that are transfected and will secrete the Vaccibody vaccine molecule. And that means that we will have the effect locally on the same antigen-presenting cells as the vaccines are targeting. And so far, the preclinical data look very promising. We have tested multiple different cytokines. One example here within oncology is where we see a 3-time increased T-cell response after a single vaccination against these neoepitopes. When it comes to the T-cell responses, that also translates into improved anti-tumor efficacy. We also see that we can use this principle within infectious diseases, and with multiple different cytokines in the 4th module, we still see a 10-fold higher antibody response compared to the standard Vaccibody molecule, and really reducing the dose of the vaccine in vivo studies. We also, for these infectious disease programs, do see a very strongly increased T-cell response at different levels with different cytokines. So we have the opportunity to include different molecules in the 4th module that all will add on to different extent on the gas pedal or the brake or the steering wheel in the future for this technology. Next slide, please. Last week, we also presented the first glimpse of data that's been generated with the platform for use within autoimmunity. So this is where we can create a tolerizing vaccine where we target specific receptors on tolerizing antigen-presenting cells. And we can also here use the 4th module principle and include immune inhibitory cytokines And so far in some of the data that we generated, we really see an increase in the IL-10 versus interferon gamma ratio compared to standard vaccines. And we see an increase in antigen-specific regulatory T cells as well in proliferating regulatory T cells, which makes this very promising for a new platform within a new therapeutic area for Nykode in the future. Next slide, please. Briefly look at the financial situation in Nykode. If you go to the next slide, we just announced the financial numbers from Q1, and we are well positioned to grow and execute the company strategy over the next years. We do have currently $238 million in liquidity, with that also includes the milestone payment of $20 million that we received first quarter based on a milestone that we met in the second half of 2021. So next slide, please. So next is the last slide. We do have now multiple programs ongoing. Obviously, we have a lot of focus on VB10.16 now after seeing the positive interim clinical data. We are looking forward to further update you on that program and also the expansion into additional indications, as well as a further development strategy for VB10.16, in addition to the work on infectious diseases, manufacturing, and technology. So next slide, and thank you for listening. We are open to take questions.
Yes, thank you very much, Agnete and Siri, for the nice presentation. And now we have time for a few questions. And I will start with my first question, would be around your recently announced data on the CO2 program. And the data seems very promising, specifically compared with checkpoint inhibitors monotherapy. And my question is, assuming that the full data set looks similar, what would be the next steps in the development process of the vaccine? Are you planning to run a confirmatory trial straight if the data points to the right direction, or you would have to do a smaller step before running for confirmation?
Yeah, no, it's a good question. And first of all, these data were very promising for us. So we are now in a phase where we need to sit down and really evaluate the next step for the program also within cervical cancer. As you see, we just announced that we will initiate a study in head and neck cancer in order to broaden our efforts with this vaccine. But for cervical cancer, certainly it will be of a high priority for us now to really discuss with regulatory input as well as other key opinion leaders what would be the optimal next path forward, and that's something we will update you on within the next months.
Yeah, thank you very much. And a follow-up question on this. You are announcing now that you will move towards head and neck cancer. Could you expand a little bit on the patient population that you are trying to aim? Are you planning to also include PD-L1 positive as well as negative patients?
Yeah, so you're right. So what we have announced is that we will move into head and neck cancer with our vaccine in combination with a checkpoint inhibitor. But for now, we haven't disclosed either which patient population and or which checkpoint inhibitor this will be relevant for. So we will come back to that within the next couple of months.
Okay, clear. And one final question before we finish. It's regarding your newly announced vertical in your company, which will be immunotolerance. Could you give us a little bit of insight on why are you choosing specifically immunotolerance? Is it a space where you really need this extra push with the fourth module that you probably don't need in a space like, for example, cancer, or what's the rationale behind that?
No, good question. I think, most of the treatments for autoimmune diseases at the moment are not antigen specific and are inhibiting the immune system in general, which also has some unwanted side effects. So there is definitely a need and a scientific rationale for work with antigen specific immune tolerance. And I think from the start, just having something that targets the antigen specifically to, for instance, tolerogenic dendritic cells is something that we find very promising, and that's something that's very close to what we have worked on for the stimulatory vaccines, and gives us a huge benefit in order to pursue that field in a more directed manner than the other efforts that we've seen so far within antigen-specific immune tolerance. And then working with the fourth module on top of that, we see that we have even one step further ability to direct immune responses in the direction that we want and see. So I think we have two steps here that adds on compared to what we've seen in other vaccine technologies for immune tolerance, that gives us a very interesting basis to pursue this field in the future.
Very, very interesting. Agnieszka and Siri, thank you very much for being here today with us and for your nice presentation.
Thank you.
Thank you.
Welcome back. The next company to present is Gesinta, and I'm joined by their CEO, Patrik Stenberg. Gesinta is a Swedish unlisted company developing novel treatments for inflammatory conditions with a lead candidate in phase 2A study for digital ulcers secondary to systemic sclerosis, an orphan disease. I'll hand the word over to you, Patrik, for the presentation.
Thank you very much, Adam, and thank you very much for having me here. I really appreciate the opportunity to present to you Gesinta today. So I'm Patrik, and at Gesinta we are developing anti-inflammatory drugs, and these inflammatory drugs also have vasoprotective effects. So that's the whole idea behind Gesinta. And to that end, we are now developing a new class of drugs which are interacting with a unique target. And by inhibiting this particular target, we are simultaneously treating inflammation, protecting blood vessels, and also relieving pain. And we're using these properties to build a pipeline around several diseases with a high unmet need, but also a great commercial potential. And to that end, as Adam mentioned, we are now developing at the forefront GS248, which is a small molecule in phase 2A. And we are now in a phase 2A study aiming for top-line data later this year. And this, again, is for an orphan disease, which is in contrast to our other compound which we're developing, which is GS073. We're a little earlier there. We have just concluded the preclinical safety package. So we're ready to enter phase 1. We expect to do that within a year or so. Here we are instead addressing somewhat different plethora of diseases, for example, endometriosis and other chronic inflammatory pain conditions. Gesinta is run by a very experienced team. We have experience in drug development, business development, and I think above all, we are all experienced pharma managers. And we are backed up by a series of investors, spearheaded by 3 VC companies, and a number of private investors with long experience in the life science field. So that's Gesinta. And as I said, we are focusing heavily on systemic sclerosis, and GS248, our front runner here. So systemic sclerosis is an autoimmune disease. It's a chronic disease characterized by inflammation and diffuse fibrosis in the joints, internal organs, and vasculature. And in particular, what we see in this patient population is a dysregulation in the smallest blood vessel, the microvessels. And so this is a disease where we see mostly women being affected in their midlife, meaning that it has a very high impact, not just on the individuals, but also on the society as a whole. And one of the manifestations of this microvascular dysfunction is digital ulcers. So these are severe, debilitating ulcerations which these patients get on their toes and fingers, and even their noses in some cases, which is actually the major cause of concern for their daily life. So it's a highly debilitating condition that they have. Of course, microvessels are also elsewhere in the body, and we also see lungs and kidneys being affected in this patient population. And what we see is also that the treatments available today are either not effective or they carry severe side effects. And in particular with digital ulcers, here we actually have nothing approved in the US and only 2 drugs approved in Europe. So turning a little bit into how the microvascular dysfunction causes these problems in systemic sclerosis patients. I have included 3 examples here in the upper panel. You can see the numbers within parentheses here being the prevalence. So you can clearly appreciate here that pretty much all these patients will suffer from some form of microvascular dysfunction, with secondary renal phenomenon being the most common one, affecting pretty much all these patients. And I will note also that this is very different from primary renal, which a large number of people actually have without needing treatment. In this patient population, the attacks that these patients suffer here are longer, and they are also more severe than normal renal attacks. and the reduced blood flow that we see here persists for a longer time, to such an extent that in about half of the cases, they also result in ulcerations on the fingers of these patients, seen here in the middle picture. And again, here we see a very debilitating condition with few, if any, pharmaceutical treatments available today. And as I said, we also see these microvascular manifestations in other internal organs. And the example here is in the lungs in the form of pulmonary arterial hypertension, which also affects an appreciable part of this patient population. And the mechanism behind this is very tightly related to the inflammation itself. So we see that the chronic inflammation, the systemic inflammation that these patients are suffering from is also affecting the vascular walls, resulting in dysregulation and of the blood vessel constriction, and blood clots and remodeling being also a result of this. And collectively, this reduces the blood flow to these patients' small vessels and causes these problems. So just a mechanistic note here in order to understand how Gesynta Pharma's drug candidates actually work in treating microvascular dysfunction. We need to take a step back and look at one of the cascades responsible for driving inflammation in the body. What we see here is a cascade whereby inflammation causes release of arachidonic acid from the cell membranes, seen up here in the gray triangles, and arachidonic acid is then converted through cyclooxygenases into an unstable prostanoid intermediate, and then using a series of terminal synthases, two of which are shown here, producing bioactive lipids. And these are compounds that have various functions in regulating the body. In inflammation, the main driver is prostaglandin E2, seen here in red. And this is usually the bad guy that we want to get rid of when we treat inflammation. And this can be done by inhibiting cyclooxygenases. There are very common drug targets being addressed by NSAIDs. And so we know that when we inhibit cyclooxygenases, we take away all synthesis of prostanoids, and including PGE2, and we get very effective treatment of the signs of inflammation, such as pain, redness, swelling, and so forth. Unfortunately, what we also do is block formation of all other prostanoids. And this in particular, in this example, includes prostacyclin. And prostacyclin, seen in green here, is one of the body's most potent vasoprotective compounds. So prostacyclin causes vasodilation, and it also causes less stickiness of the thrombocytes. So it's also antithrombotic. Now, of course, we're all aware that when we inhibit cyclooxygenases, in particular cyclooxygenase 2, we are seeing a fair amount of cardiovascular side effects, which is to be expected because we're also taking away prostacyclin. Now, this is in contrast to Gesynta Pharma's drug compounds. What we are doing is that we are inhibiting an enzyme further down in the cascade, here denoted as MPGS1, or microsomal prostaglandin E synthase 1. So this is an enzyme which is responsible for synthesis only of PGE2, and also primarily during inflammation. So it's an inducible enzyme. There's not much MPGS1 around when there is no inflammation. And it's been known for quite some time that from preclinical studies that when we inhibit MPGS1, we are also taking away PGE2, reducing signs of inflammation such as pain and swelling. What is new here and what was discovered by our scientific founders is that when we inhibit MPGS1, we not only take away PGE2, which is the bad guy here, but we're also, in this particular case, reverting the synthesis of this unstable prostaglandin intermediate seen in blue here over to the prostacyclin pathway. So we get more prostacyclin. And of course, prostacyclin being vasodilatory and antithrombotic, we are also achieving a vasoprotective effect. And this is something we have seen preclinically in several models with human blood vessels even. So this is how we can, by hitting one target, achieve both an anti-inflammatory and a vasoprotective effect. And in a disease such as systemic sclerosis, which is caused by inflammation, but then again manifesting as cardiovascular problems, this is particularly well suited because we're benefiting from all the three arms of this treatment. So this is all well and fine. Preclinically, we have a very solid case, as I said. It's been demonstrated by ourselves and others that this mechanism actually works. So in the first human study we did, the phase 1 study, that was actually 2 years ago now, where we studied GS248 in healthy volunteers, we did not only include the traditional safety, tolerability, and PK parameters, but we were also able to study formation of these prostanoids. And so in the left panel here, you can see the formation of prostaglandin E2 in these subjects. And in the graph here, you can see that we have administered placebo 20, 60, and 180 milligram of GS248 as one dose. And it's also quite evident here that in all doses here, 60 milligram and upwards, we have complete inhibition of PGE2 formation in these subjects. And I will also say that these were samples taken 24 hours after administration of an oral dose of GS248. So clearly a good pharmacodynamic profile here. And the other side of the coin, we have prostacyclin. And in the right panel here, we show the excretion of a stable prostacyclin metabolite in the urine of these subjects. And here we have a converse relationship where we can see that in light blue here, the placebo group, we don't see much difference in the excretion, whereas in the 20, 60, and 180 milligram groups of GS248, we see a 2 to 3-fold increase in formation of prostacyclin metabolites, thus also showing that this shunting mechanism postulated from preclinical data also works in humans. And as a control here, we have included celecoxib, which is a COX-2 inhibitor, which is doing exactly the opposite of an MPGS1 inhibitor, i.e. we are actually decreasing prostacyclin, which is also in line with what's being observed in the clinic and also, of course, the hypothesis. So in summary, this first study, being a phase 1 study, we could still show that we are inhibiting PGE2 production very potently. We actually have a potency of less than 0.5 nanomolar in humans here. And we are at the same time increasing prostacyclin, which is a very well-known mediator of vasoprotection. So this gives a very good potential for GS248 and other MPGS1 inhibitors to actually be vasoprotective and anti-inflammatory. Other things, of course, that we study, we're able to support that we have a very safe and well-tolerated treatment, and we're looking at clearly a once or twice daily oral dosing and clear differentiation from NSAIDs here, such as celecoxib. And this data prompted us to go into the phase 2A study, which we mentioned initially. So this is an exploratory study in systemic sclerosis patients suffering from Raynaud's condition. Here, we have a 1-to-1 placebo-controlled study where we're administering single oral doses for 1 month of GS248, 120 milligram. And we're doing this in several sites across Europe right now. The interesting parts of this study is not, of course, just to study the safety in a systemic sclerosis population of MPGS1 inhibitors, but also to study a number of parameters related to vascular function and inflammation in this population. So to that end, we have several efficacy endpoints to understand what we can do with an MPGS1 inhibitor going forward in the clinical program here. So, and I will also say that, of course, we are also having a mixture of objective measures, such as with measuring the vascular blood flow, we're measuring biomarkers of inflammation and vascular status, but we're also measuring subjective parameters such as Raynaud attacks, pain, and how severely these patients are affected by Raynaud syndrome in their daily life. So the program right now, as it stands, so far we have completed a number of phase 1 studies. I show you the main one here, but we also have done several clinical pharmacology studies to study interactions and with food and other drugs. We have an open US IND, and we just recently got an orphan drug designation granted in the US. And we have the ongoing phase 2A study, which I mentioned. And the next step is to study in a longer duration. We expect this to be a 4-month study where we will study both Raynaud phenomenon, but also development of digital ulcers. And then, of course, long term, we're looking at 1 or 2 phase 3 studies to allow for registration within systemic sclerosis. I will also just say a few words about the competition that we're facing here in this space. As I said in the onset, we have actually for digital ulcers, which is what you see in the table here, we have nothing approved in the US. And I'm showing the table just the two products which are approved in Europe. One being iloprost, which is a prostacyclin analog, and which you may recall also from the flow chart of prostanoid metabolism here. Also that where I claim that prostacyclin is very good for these patients. It's very clearly evidenced here because that's one of the drugs which are being used to treat systemic sclerosis, in particular digital ulcers. Unfortunately, iloprost is a very unstable compound. It also carries a severe risk of tolerability issues. So it's being given as an as an IV infusion under very close medical supervision, and these infusions takes about 6 hours and has to be repeated several times a week. So clearly not something which can be used on a regular basis for these patients, but more as a form of crisis control. Conversely, bosentan, the other approved drug here, is an endothelin receptor antagonist, which is oral, but at the same time, according to EULAR, which is the organization's issuing a treatment recommendation here in Europe, it can only be given as a last resort when everything else has been tried, and this is due to fear of hepatic toxicity in this patient population. In contrast, the target product profile of GS248, of course, we're vasodilatory like the competitors here, but we are also inherently anti-inflammatory. And to that end, we believe we are unique here from an efficacy standpoint. We also see potential for pain-relieving effects, which we don't expect from these other competitors. And of course, no safety concerns seen so far. And this will be, again, an oral product. So wrapping up the pipeline here of what we have now the foremost candidate here in systemic sclerosis, GS248, which is also being investigated for other vasculopathies, such as PAH, which has a very similar etiology. And then we have GS073, which, again, as I said, we're looking at broader populations here, little different type of indications. We are still benefiting from the pain-relieving, anti-inflammatory, and vasoprotective effects, but we are looking into inflammatory pain conditions and endometriosis for this one. The team behind Gesunta, we mentioned that we are a very experienced team. We also being supported by 3 very good VC companies, which are backing us up with advice and others. And that is also, of course, reflected in the board composition. So we are a very experienced team, we believe. And we also have a good track record to back that up in terms of delivering clinical and preclinical data in a very consistent manner to bring us to the point where we are today. And looking ahead, we see several opportunities to create value here. With the pipeline I just showed you, we're looking at readouts from 3 phase 2 studies within the next couple of years. And we're also looking, of course, at several other more minor endpoints, but still very important in order to progress these programs. And this is my last slide, actually. I just want to wrap up that I hope I have been able to convince you that we have a very, very interesting drug target that we are addressing here, and that the opportunities of being able to not only treat inflammation, but also to have a profound vasoprotective effect is creating a lot of opportunities within several disease areas. And with that, I would like to just conclude by saying that we are expecting the phase 2A data from the systemic sclerosis study to read out sometime during the fall, I would say September. And on the back of that, we will be seeking funding for the next step of our journey here.
Perfect.
Thank you very much.
Great. Thank you very much for that presentation, Patrick. A few questions then. You showed that slide with how GS248 compares to the available treatments, which are suboptimal to say the least. But within this sort of space of fibrosis, inflammation, there is obviously other drugs in development, some targeting arachidonic acid pathway, some other pathways. Mechanistically, how differentiated would you say GS248 is from I guess the field at large?
I would say it's quite unique in that we are addressing the vascular components, because obviously these patients are suffering generally from fibrosis, and the fibrosis will also cause in and by itself problems, for example, with the lungs, such as PAH and interstitial lung disease. There are several developments in that area, none of them which are actually specifically addressing the vasculopathy or the microvascular dysfunctions. I would say there we are unique. We are actually alone there, I would say. But of course, it will spill over, and we also see in this space an increasing use of combinations of drugs. So I don't see that one would necessarily outcompete the other here. And we see that in particular in PAH, where pretty much everything is combinations.
Okay, oh, great. And within specifically digital ulcers, then secondary to systemic sclerosis, we have these two suboptimal drugs. How should we be thinking about GS248 in terms of the positioning and also pricing relative to these drugs?
So I think pricing-wise, we need to remember that these drugs were developed primarily for PAH, right? So, and there the price point is expected to be higher. What we see today with novel drugs coming in today for PAH, we're looking at treatment costs of roughly $100,000 per year. Some of them are a little more expensive, like Risoglutat. Some of them a little less. So in our pricing models, we have been using between $20,000 and $60,000 per year in the US, and of course less in Europe, depending on which country we're talking about. So definitely the pricing will be less. Positioning-wise, I did not speak of that, but there are of course off-label use in this area, and a lot of these patients are currently on calcium channel blockers and PDE5 inhibitors such as sildenafil, right? And actually in the phase 2A study, we're actually allowing those. So we are treating on top of what is being used today. Because the current standard of care, as we know, is so inefficient. It's only a fraction of the patients which deem their current treatment to be sufficient. So I would say it's about 80% of the patients where there is room for one more on top. So we're not looking into getting into first line here in the first instance, but rather second or third.
Okay. No, that's helpful. And maybe just one last question then. In the study that you plan to do, you're looking at the development of new ulcers, new digital ulcers as an endpoint. How should we think of that endpoint in terms of it being potentially liable to external factors, trauma to the hands, and these things kind of skewing the data and making it, I guess, more risky in terms of the reliability of getting good efficacy there?
Yeah. So this is what I would call a good question because it's difficult to answer actually. So I will say, and I will not speak about that, but we are having quite extensive dialogue with the FDA with respect to the endpoints of that study. Partly because we're looking for the study to be supportive for a registration. I'm not saying it will be, but we're looking for it to be that. So we need to understand the patient-reported outcome or the clinical outcome assessments. but also, of course, what are the factors that will sort of be systemic sclerosis specific to development. But I will say that these ulcers on the fingertips are pretty much exclusive to the vasculopathy, as opposed to ulcers that occur more proximally, like on the knuckles, for example. These are typically ulcers you can get by just bumping into a door. So by limiting the readout to the tip of the finger, we believe that will probably take care of that. But there will, of course, be a need to have a rather extensive study in order to factor these things out. So this is going to be a fairly extensive study. We're looking at least 10 countries and maybe 30 study sites in order to get the numbers up.
Okay, Patrick, I think we're all out of time. Thank you very much for a very interesting presentation.
Yeah, thanks for having me, Adam, and thanks everybody for listening in.
Hi, and welcome back. Our next company is Xspray Pharma, a Swedish biotech company developing improved versions for marketed cancer drugs. With us, we have here today Per Andersson, the CEO. Please, Per, the floor is yours.
Thank you for inviting us. My name is Per Andersson, the CEO of Xspray Pharma. I will tell you about what XP is all about and where we are at the moment. But we're working with improved protein kinase inhibitors. And let me just say that we see the human as a body with a hollow tube inside it. This hollow tube is having very many conditions. It's wet, it's dry, it's acidic, it's neutral. It goes from the mouth and you know where. This means it has a purpose of serve and protect its intestinal system, and it's actually outside the body. And the question is how to get the pharmaceutical into the body when you actually have an oral administered drug. A drug that comes out to the market is not always readily developed, finished product. After a couple of years, you start to realize what the tablet is missing in order to get over this obstacle course. Now, X-Spray is making it better and improving that part of the product, actually getting the more reliable absorption into the body, and that's what we're going to talk about today. So we develop a technology to get drugs better absorbed into the body, and we do this with something called protein kinase inhibitors that we concentrate on. It's more than 25% of the total sales of cancer drugs. We're focusing on the US market. We are building a powerhouse of improved PKIs, protein kinase inhibitors. This inhibits the tumor growth, the cancer growth. It doesn't always cure it, but it inhibits it, and it's one of the reasons why cancer drugs, why cancer is transforming from a deadly disease to more of a more and more a chronic disease. Chronic disease also means that you have improvement of quality of life, health-related quality of life issues, and that's exactly where we are focusing on. It's all about the quality of life for patients. To build this powerhouse, we have our patented technology. We have a unique in-house patented technology that is a sort of foundation for the whole thing. We have the US as a primary market. It's a solid and world's largest market, and we understand it's the patent scenarios, the patent rulings, and the regulatory parts. One while going to other markets is, of course, after we have got the launch going in the US. We have the first product under review at the FDA, and I'm glad to say that in terms of company-wise, we need sort of no learning on the job when you come to this type of late-stage development. So I have some of the most senior and most well-developed senior people for pharmaceutical development in Sweden. It's a true joy to go to work in the mornings. The business model that we're working with, because we have a unique technology, we also have a unique business model to some extent. The uniqueness here is based on the fact that the tablet that has been developed, the originators has their income from a patent life. And when that patent expires, a generic entry come in and are the first competitors in line. In some cases, the originator company managed, or actually in 25% of the cases, the originator company managed to bring in secondary patents. That prolongs the lifetime of the product. And hinders the generic from entry. Now, if you have developed a unique technology that we have, there is possibility to actually enter the market and improve patients' life earlier if you're not affected by the secondary patent, and still being semi-exclusive then with only very few or only one competitor, the originator company. So in this way, we can improve life for patients and get in early to a very well-established and high-value market. We can do this because we have this unique technology platform, and the market for these drugs they have very good health economics, so they're very highly priced. It's orphan drugs, and in this case, we come in with an improved product, and we actually improve then first-line therapy, or it's rare that you can actually have the have the chance to affect the standard of care of first-line therapy. But here we truly have a chance to actually do that. We also have the possibility for early access during launch window. And when we go in for a blockbuster product like this, improving it, we know that we need freedom to operate towards this secondary patent. And in this case, for dasatinib, our first product, for example, we have 2 different law firms that has given us written consent that we have freedom to operate. Of course, we expect to get sued at all products that we put in there, because that's the way it works. Given that, we're also covering our back, of course, with our own patent portfolio. That is based on technology and also of composition patents for the products that we developed. If we look at the technology, if you Google supercritical fluid antisolvent precipitation and you get the review article, you will see that this technology to make particles, the pharmaceutical active ingredient as a particle, was introduced in life science or all the large pharmas in the 90s. Now, there's no product out yet, and that's because no one could scale it up. So our claim to fame is that we can scale it up, and for that you need a unique nozzle head and you need a big machine. And we have both of them together now, and it's been running a couple of years, and we know that we have a stable manufacturing technology in 2 lines operating right now. And this has been a very important step for Xspray to get this established, because that's a foundation for the company. Now, if you look at the products and with what we are doing, on the left-hand side here, you see the crystalline version of a compound coming in. That is the originators, not the originators always, but it's a crystalline version. When you put X-ray on it, you get a fingerprint. We convert it in our system to an amorphous, non-ordered version. It's amorphous, and it becomes completely different in terms of when you try to look at the solid state. It's not crystalline, it's something else, it's amorphous. So it's not even relative family with crystallinity. And that makes also a good argument for in the patent type of litigation part. So we know that we're not infringing in that terms, and what that amorphous form does is that if you look at the next on the slide with the human bodies here, you can see how the crystalline version has a lower solubility than the amorphous one, which means that it processes issues when having the stomach as the distribution channel. And it is important to get as much of the drug into the body as possible, because otherwise you risk adverse effects. And in our case, we reduce the dose that is swallowed, but it's at least as much that actually gets into the body. Okay, so we have the same pharmacokinetic profile. We get the same amount into the body as the crystalline, but we use a lower dose. We have less stress on the intestinal system, which can lead to less adverse effect and a better uptake. That will be, and I should say that also that in terms of having stomach as a distribution channel, if it's acidic or neutral for these protein kinase inhibitors, is very important. Many of them are very sensitive to pH on how much actually enters the body. And that is, for our first product, something that we address. You can't have peptic ulcer then you should avoid the cancer medication. But of course, you need the cancer medication, so you rather avoid the antacids. We look at the product portfolio of 72 protein kinase inhibitors in the US. 58 of those are polysoluble. They're in all fields of cancer, all sort of indications. We are targeting something called chronic myeloid leukemia, first of all, CML. It's a blood cancer disease. We have 2 products ongoing in that. But in reality, if we look at our 72 possibilities here, It's actually 16 that goes out of patent into 2027 that we're currently addressing, and of those 7 is meeting our criteria of sales and patent launch window and improvement part that we can do. So we have 7 products in our portfolio right now. We have the first one, Dasinoc, is based on dasatinib, Sprycel, blood cancer, CML. We have conducted the clinical studies successful. We have a label change in it, so we know that we're gonna get an improved product out there, and we know that it's we certainly know that it's in FDA for review right now. We receive questions from them, and we respond just like normal, and we expect this FDA review time to end in a tentative approval late this year. If we look at our product pipeline, we have Dasinoc that has progressed the longest. In hematology, in the same area, CML, we have Hinapnilo. Hinapnilo is an really interesting product. It is the only product on the US market with a food interaction label, as if you basically say if you take this with food, you might die, because the side effect is called sudden death. So it's a drug where you need to fast 6 hours per day. And of course, if you can remove the food interaction of this drug, then you have a severe improvement. And we have managed to do that in the first stage, and now lately have repeated some of these clinical studies with Hinapnilo, so that is progressing according to plan. The patent window there, or launch window there, starts in January 2024. If we look at the other parts, you might wonder why we still don't disclose our portfolio that we're working with. I mean, the technology is good for all PKIs that we have tried with to date, which is more than 20. So we have 7 in our portfolio right now. We're not disclosing which ones, except for these, because of competition, both from the originators but also from other competitors. So you have to wait for us to do that. We do that in time. But we have products to take to the market, and then come the question about the market and the market potential and how to commercialize. And if we look at the market potential, consider Bristol, the first product, Dasinoc. It sells for $1.3 billion in the US yearly. That is $100 million plus a month. If we can enter that market and say that we take 10% of the market and have the same price as Sprycel, then we would, you understand which type of sale we would achieve. We can see that biosimilars can take 50% in a year. We'll see where we can go with this, but there's certainly a huge market potential with the type of products and the improvements that we have. And after US, I would say that we're planning to expand to other markets in the rest of the world, but the US is a primary market. In terms of, we expect litigation for all the products that we do. That is the defense of the originator. So we always have that from the start when we start building our system, we're building our project. And it is a natural part of the business in the US to get sued, and we are under litigation in terms of the first product. In the terms of the FDA approval process, we believe that after we have the first one done here in the end of this year, that sort of paves the way also for the next and the next one of our products. It becomes easier for us when we know what they want to know and how things looks like. So I think that will ease up on us and lower the risk. In terms of commercialization, there are several paths to go. We have partnering discussions with either licensing out the product or commercialization specialists. There is sort of a valuation part of this that we have to consider, considering whether it's 2 products in hematology. Would you license out one and keep one by yourself? Would you see it as a portfolio and then regard the rest of the product as a new portfolio? So we're currently under discussion and determining the commercialization. In the meantime, we're building a solid pre-launch program in-house. We have a commercialization head just hired. We have CMOs coming in. So right now we're in the middle of the commercialization process. The tricky part is to know when the start point will be. We expect it to be in the second half of 2023. And to summarize, we have a unique technology, and that allows us to have a unique business model with improved products in a very highly valued market, and where we have a good chance to improve quality of life for the current patients. And the key takeaways that I want you to have is that we have a patented and scalable technology where we improve we can, after many years of these products on the market, see what the obstacles issues has been in history. Design the tablet the way so that you have an improved version coming that it improves life for quality of life for the patient. We have the first product under FDA review. We have a solid, promising pipeline coming up. We have US as primary market because the size and the ease of, for us, relative ease of access. And I believe that with the type of competence that we have gathered in the company, I feel really good for the future. And with that, I would like to thank you and open the floor for some questions.
Thank you very much, Per, for such a presentation. And now we have time for a few questions. And I will start giving you, asking you about what are the commercial benefits that bringing an improved version to the market has compared to a normal generic?
That is a big difference. The normal generic has to be the same as the originator. And if you are the same, show to be the same, then you will also have the same disadvantages. So when the patent expires, when generic entry comes in, if we have established a significant improvement with the patients, we will also withstand that patent cliff better. So we will have value. I mean, if we, for example, you can't take antacids. For the generics, you still can take antacids, while with ours, the patient can take antacids and still get the cancer drug absorbed. So you still have an effective drug and eat cancer. And we know that more than half of the patient population in this field needs antacids. So we believe that in that respect, we will have a good patient group to work with. The disadvantage is that the uptake on the market, now we're going to have to sell the improvements to the doctors, while generics are substitutable. So it's a much steeper uptake in the market of substitutables. But on the other hand, we can say how different drugs, for example, biosimilars, is taking 50% of the market within 1 year. So that difference is it's going to be very interesting to see what type of how steep we can get it and what penetration we can achieve. And I think that is the whole purpose of the commercialization effort that we're doing right now with the pre-launch program.
No, it's very interesting. And now you mention it, and that you will have to bring your product to doctors. Do you have any defined strategy on how to do that, how to reach doctors and convince them that Dastinoc is what they should prescribe?
Yes, and that's what we're building. So what we need to do right now, because of time and have started, is to build a solid pre-launch program that then can be fitted into the partners. So we're not building our own sales organization at this stage. We're going to click in that part into the partners that we choose. And that partnership is chosen then in terms of the full portfolio in consideration.
Okay, very, very interesting. And my next question is regarding Europe. Right now you are tackling the American market. Do we have any kind of timeline in the midterm that you will try to reach Europe, or it's still far from now?
I think it's very Europe is a little bit different than US, and that's because of patent. For example, dasatinib doesn't have a patent, a valid patent for the substance in that was invalidated a few years ago. So it's a very different situation in terms of patent life. Now, generics for dasatinib is getting there, and in Europe, and establishing a lower price. Whether we can see that our improved version would work here at a higher price or at the same price, we can consider Europe as a market. It depends on how well, what the uptake and the need is that we see in the US. But for nilotinib, there's still a patent window also in Europe, and we're looking forward. We're of course investigating the different market approaches after the launch of the US product.
Okay.
But it certainly are possibilities for the other markets. Okay. But they're different because of patent situations.
Okay. And my next question is for both products, for Hynapnilo and for Dasinok. Do you have, assuming an approval by the end of the year in the US, do you have a strategy, for example, in terms of commercialization, in sales for Dasinok, but for all your portfolio, and how are you planning to tackle this?
I think that we need to address the healthcare professionals in one field at a time. So right now we have 2 products in the hematology portfolio, and you can see we had one for kidney liver as well. So if we start with we addressing the oncologist and the hematologist right now, and then at the later stage we gonna go for, when we start the other products, we go for that those specialists, so to speak. So it's a specialist sale. These doctors, these healthcare professionals are amongst the most well-educated that we have. So it's gonna be it's a well-educated sell as well, if you see what I mean.
Yeah, I understand.
It's very data-driven.
Okay, interesting. And now we have time for one last question, and it's regarding cash. I don't know if you could remind us what is your current cash position, and if needed, how are you planning to finance yourself given the current market situation?
The plans for our financing situation is dependent on the partnering discussion and our expansion plans. Right now, we're fine. So it's just a matter of which speed do we want to develop. I have more projects than I have resources.
Yeah.
So it's all amongst how much do we want to accelerate now when we know that it works. So we're gonna see for the FDA review now and get the first product approved and partnered, and then I'll let you know.
Per, thank you very much for the nice presentation and for being here today with us.
Thank you.
Hi. Our next company is Bergen-Bio, a company developing innovative drugs targeting aggressive diseases such as cancer and infectious and respiratory infections. With us, we have here today Martin Olin, the CEO. Please, Martin, the floor is yours.
Thank you very much for the introduction. So I'm Martin Olin, the CEO of Bergen-Bio. I'm very pleased to provide you with an update on Bergen-Bio plan. If you can click to the next slide, please. So I'll just remind everyone about the forward-looking statements. And we can flip to the next slide, please. And click again. It should be, yeah, thank you. So Bergen-Bio believe that AXL as a target is a very interesting treatment modality for a number of severe diseases. We consider ourselves to be the world leader in exploring AXL as that transformation treatment modality for severe diseases. Click, please. I'm sorry for the animation. Our lead program, bemcentinib, which is a small molecule, orally available and highly selective for AXL, has been dosed in more than 600 patients as of today, about 400 in oncology and 200 in respiratory diseases, specifically COVID-19. And we are now advancing bemcentinib into two significant indications, being one, first-line STIK11 mutated non-small cell lung cancer, and secondly, hospitalized COVID-19 patients. Click again, please. We are applying a laser-focused approach to unlock clear value drivers within the next 18 to 24 months linked to those two specific indications. Click again, please. And Bergen-Bio, in pursuing these two indications, retain all the rights to our candidates, positioning us well for potential partnering going forward. Next slide, please. So the broad potential of bemcentinib has been extensively explored within a number of indications and shown clinical efficacy. I'll just remind the audience that bemcentinib's mode of action is blocking AXL activation, which we know is relevant in a number of diseases, primarily in respiratory diseases where it's relevant in viral entry, cofactor, immune suppression, the ECM production, migration, and basal cell proliferation, but also in the cytokine response. In cancer, it confers when AXL is activated to invasion or migration, drug resistance, proliferation, survival, and immune suppression. Click again, please. So this is a snapshot of all the indications where bemcentinib has been tested. And there should be a tick mark actually on AML. So we are, of course, very encouraged by the fact that in practically all the indications, except for pancreatic cancer and glioblastoma, we have shown clinical efficacy of bemcentinib. Next slide, please. This significant clinical exploration has also allowed us to understand the properties of bemcentinib. So we know that it accumulates very well both in brain and in lung, which is particularly relevant to the two diseases in our focus area, which are both lung-related. It also crosses the blood-brain barrier, which may be particularly important for treating metastasis. We have done an extensive analysis. As I said, of bemcentinib, we understand very well the pharmacodynamics and the pharmacokinetics of the compounds. We understand the interaction with other compounds. We've done a number of combination studies. We also understand very well now the interaction with food and fasted stages. And we believe that we have been able to define dosing regimens that defines an acceptable balance between safety and efficacy. Next, please. So the rationale for first-line STIK11 non-small cell lung cancer, why is it so? First of all, it's a very significant portion of first-line non-small cell lung cancer patients that harbor the STIK11 mutation. It's about almost 20%. and if you can click. And that specific population, which I said is significant, does not respond very well to checkpoint inhibitors. Click again, please. And we've also shown, very interestingly, that benzenzimab is actually able to restore the activity of immune checkpoint inhibitors, which we have demonstrated both in preclinical studies, but also in early clinical development. Next, please, click again. We believe that benzenzimab is the only highly selective AXL inhibitor in development for this specific population. And we also believe we have a strong proprietary position with IP filed in this area. Click again, please. We have received a fast track status from the FDA in this specific population. And if you can click again, please. We believe that given the unmet medical need, there is a strong argument for an accelerated approval pathway in this specific population. Next slide, please. This is a snapshot of the today's existing treatment regime for treating non-small cell lung cancer patients. So one could simply say that the treatment regime depends on whether you have a molecular-driven disease or whether you have a non-molecular-driven disease. So in the non-molecular-driven situation, standard of care today is immune checkpoint inhibition combined with chemotherapy by large. But if you have a molecular-driven situation, which is actually a significant portion of non-small cell lung cancer patients, those patients are treated with targeted therapies like EGFR, ALK, and BRAF inhibitors. But there is still a number of molecular-driven situations which today does not have any good treatment options, and a significant portion of those are patients who harbor the STIK11 mutation or mutation in the gene of the STIK11, and it has been reported to be up to 20% of first-line non-small cell lung cancer patients. Click, please. So we do believe that given the preclinical activity, but also the clinical activity and the mode of action of benzenzimab, there is an opportunity to create a specific and very significant patient segment that could benefit of the treatment from benzenzimab. Next slide, please. There are multiple sources that confirms that STIK11 mutations are associated with low response rates, but also short median overall survival when treated with immune checkpoint inhibition in combination with chemotherapy. Okay, please. So in general, it has been reported that the response rate to immune checkpoint inhibition in STIK11 mutated patients is around 3% or definitely below 5%. Click, please. And we also see a significantly shorter median overall survival for STIK11 mutated patients compared to the wild type population. There's a more than factor 2 difference, suggesting or pointing to a significant unmet medical need for this population. Next slide, please. We believe that benzenzimab has a novel mechanism of action to generate new tumor-specific CD8 plus cells. And it's known that one of the major premises for checkpoint inhibition to work is actually the presence of CD8 plus T cells. Click, please. And we know that, as I said, that they are key to respond to PD-1 or PD-L1 blockade. Click, please. And the unique mechanism of action that benzenzimab confers to is that it increases the type 1 interferon secretion from the dendritic cells that drives the generation of a new tumor-specific CDL population, which then restore the therapeutic response to PD-1 therapy. Click, please. And we feel that this very well fits into the combination approach of immuno-oncology, was a focus to reinvigorate the effect of immune checkpoint inhibition, as shown here also on the cartoon on the left-hand side. Next slide, please. So in March of this year, we reported some very compelling preclinical data on the activity of benzenzimab in this specific population. Click, please. So in two models, we've shown that when benzenzimab is added to pembrolizumab, we see a significant increase in the CD8 T cell plus population compared to both the control and also pembrolizumab on its own, which, if you click again, also showed a significant reduction in the tumor volume in these two models. Next slide, please. We also went back to or into our second-line non-small cell lung cancer study, the 008 study, which is soon to be reported. And we actually were able to identify 4 patients who harbored the STK11 mutation, and as shown on this slide, all of these 4 patients had good responses and have also had a significant extended duration of treatment compared to the wild-type population. Next slide, please. So the path forward in STK11 for BerGenBio, specifically non-small cell lung cancer, is, click please, to further advance the understanding of the clinical setting and increase the awareness of this significant unmet medical need in collaboration with key opinion leaders and academic centers, both in Europe but also in the United States. Click please. With that, we are preparing to conduct a phase 1b/2a study in the US and Europe in the second half of 2022. Next slide, please. And we're also expanding the preclinical understanding, including the mechanism of action and co-mutational efficacy, as it's known that there are several co-mutations that may or may not impact the efficacy of immune therapy, and therefore also the role of benzenzimab in this setting is critical to understand. Next slide. And I'll then move over to the COVID-19 opportunity. The rationale for COVID-19, in our opinion, is that the unmet medical needs still persist. The number of people hospitalized are stable. And I think we all need to be reminded that of those 6.2 million unfortunate death events due to COVID that the world has seen, 3 million of those happened in the last 12 months. So the pandemic is still around. Click, please. And we believe that despite of the recent approvals with vaccines and other treatment options, the medical need for hospitalized patients requiring oxygen is still there. Please click. We have shown in preclinical setting that benzenzimab is insensitive to variants, meaning that we believe that benzenzimab potentially has a broad coverage across those variants that we have seen and will continue to see, in our opinion. Click, please. We have seen a profound efficacy signal in the ACCORD-2 study, which was conducted in hospitalized COVID-19 patients. I'll come back to that. Click, please. We've also been accepted into the EU SOLDAC platform study, which will be conducted in up to 500 hospitalized COVID-19 patients, and we expect the study to accrue rapidly given the ongoing pandemic situation. Next slide, or click, please. We also believe that the regulatory agencies are expected to continue to grant accelerated or emergency authorizations for promising drugs, which we believe benzenzimab represents. Next slide, please. So the unmet medical need persists, in our opinion, and we believe that benzenzimab is likely to be effective against current and future viral variants. Click, please. This is a snapshot of the preclinical data, which clearly shows that benzenthamib provides a broad COVID-19 variant inhibition, and therefore will, in our opinion, give the same clinical benefit regardless of the variant. Click again, please. And new variants are likely to occur. This is a statement from Dr. Anthony Fauci, from the head of the NIH, and I think he is reminding us clearly that the one thing we all have to appreciate is that as long as we have a virus circling around, it's very likely that we will see new variants. So the pandemic will be around for some time. Click again, please. We also have seen that although the level of hospitalized patients have gone down, primarily due to the vaccines and the spread of the virus, so people have some kind of immunity, the number of patients hospitalized seem to be stable over the last couple of months now. Next slide, please. This is a snapshot of the ACCORD-2 data, in which benzenthamib met its primary and key secondary endpoints with very strong statistical significance. So the cartoon shown on the left-hand side of the slide shows the additional clinical benefit that patients saw when benzenthamib were added to standard of care. There were a significant reduction in the number of death events in the benzenthamib arm. We were able to prevent patients to require intubation. We were also able to reduce the number of patients who required oxygen, and also importantly, we were able to discharge the patients much faster from the hospital than compared to standard of care. So all very meaningful clinical benefits that we believe are very attractive in this setting, especially in different pandemic waves where the resources on the hospitals and the capacity is under pressure. Next slide, please. This is a snapshot of the number of sites being involved or signed up under the EU Solid Act platform, which we believe is very attractive. I think it's about 75 sites who are active this moment, which tells you that we have an infrastructure that will allow us to recruit relatively fast in a pandemic situation. Click, please. We believe also that EU Solid Act has demonstrated the ability to rapidly recruit hospitalized COVID-19 patients in previous studies. Click, please. We believe that the treatment with benzenthamib or matched placebo, in addition to current standard of care, will reflect the evolving or revolving environment of new agents. Click, please. And we also believe that the study design actually takes into consideration the nature of the disease behavior and also the effect of the vaccines and the variants, which is a very important element in this phase of the pandemic. Click again, please. We also believe that the primary endpoint that has been selected in discussion with the authorities are informed by the data from our 2 previous COVID-19 studies. Next slide, please. So the path forward in our COVID-19 focus is, please click, to demonstrate the efficacy in patients on top of current standard of care, including remdesivir and corticosteroids. Next slide, or click again, please, sorry. We believe that the EU Solid Act provides us with a unique opportunity to generate data in a large multi-site study in a highly cost-effective manner. Click again, please. The COVID-19 data could lay the foundation to explore benzenthamib in other severe infectious diseases, which will, in our opinion, further expand the market opportunity. Click again, please. We also believe that if we are able to replicate the data that we've shown in the Accord 2 study, that could be the foundation for a discussion with the authorities to expedite the route to the market. Next slide, please. This is an overview of our focus strategy on the 2 indications on bemcentinib. First, non-small cell lung cancer, first line, STK11 mutated patients. The phase 1b study, which is a traditional 3+3 approach, 3 cohorts defining the safety and potentially early efficacy expansion into STK11 mutated patients in a phase 2 dose expansion study. And in parallel, exploring and expanding the non-clinical understanding of bemcentinib's role in not only STK11 mutated patients, but also in co-mutated situations. And secondly, the respiratory infection or COVID-19 focus, which primarily is the conduct of the EU Solid ACT study in up to 500 patients, and further exploration of bemcentinib's potential role in respiratory indications by means of conducting additional non-clinical studies. Next slide, please. So in summary, please click. We consider ourselves to be the world leader in exploration AXL as a transformative treatment modality for severe diseases. Next, click. Our lead program, which is our focus going forward, has been extensively studied in more than 600 patients, and we are now moving forward in 2 significant indications where we believe that there is a strong scientific rationale, preclinical and clinical data to suggest that bemcentinib could be a transformative treatment option in these 2 indications. Click again, please. We are applying a laser-focused approach to enable us to unlock clear value drivers in the next 18 to 24 months. And last click, we retain all the rights to bemcentinib and therefore position as well to both have the flexibility but also pursue partnering opportunities. That is the end of my presentation. Thank you very much.
Thank you very much, Martin, for such a nice presentation. And now we have time for a couple questions. And the first one is regarding bemcentinib in non-small cell lung cancer. You have mentioned that 20% of these patients have STK11 mutation. And my question is, is it in terms of testing the mutation STK11, is it something that is regulated by guidelines? And if not, how are you planning to include or to manage to include this mutation in clinical veterinary practice?
So it's a good question. So it's not regulated today, but STK11, the mutation, is on the panel of several of the NGS providers. So we believe that it's becoming a routine assessment of the non-small cell lung cancer patients when they see a doctor in the hospital. And therefore, we believe that by having STK11 on the panel, we will be able to identify the patients by that mean, and therefore conferring to the existing practice of targeted therapies if you have a molecular-driven situation. We also believe that bemcentinib, in combination with an immune checkpoint inhibitor and chemotherapy, confers into the existing regime of treating molecular-driven diseases in non-small cell lung cancer.
Interesting. And I have another question in the same indication, well, in non-small cell lung cancer. You're also pursuing this therapy for these patients in the second line, independently of the mutation. How is this program going, and are you still pursuing this group of patients?
So you are referring to the 008 study, I believe, which has been conducted in second line, all comers, both in combination with chemo and pembrolizumab. Not the triplet combination, I should say, though. The study has been fully enrolled, and we are looking to mature the data and will report the data once we have the final tables, list and forms. We do believe that the Stick 11 opportunity in first line is, given the balance, is a more attractive opportunity. It's not every day that you get the opportunity to work with first line, and therefore the second line, opportunity is not a priority for the company at this moment.
Okay, thank you very much. And one last question. Could you remind us how does your cash position look like now? And if you're planning to finance yourself in the future, how is your strategy going to be conducted due to the current market situation?
Yeah. So according to our Q4 report, we had about 436 million Norwegian kroner in cash by the end of 2021. Our average quarterly burn rate is around 75 million Norwegian kroner, which tells you that we have about 5 to 6 quarters of burn measured on the historical rate. I don't think you have to be a genius to figure out that without the top line, the company will still require additional funds to pursue its strategy. We have so far been successful in securing the adequate funds to pursue our strategy. So we will continue to pursue a strategy of raising the adequate financial means to pursue our strategy, not necessarily only pursuing a capital market transaction or transactions, but also looking at partnering opportunities to support, but also accelerate and strengthen the development going forward.
Thank you very much, Martin, for being here today and for your nice presentation.
Thank you, and thanks for having me.
Welcome back. Our next company to present is Isofol, and we're joined by CEO Ulf Junelius. Isofol is a Swedish late clinical stage oncology company, market cap around 1.5 billion Swedish, developing its lead asset arfolitoxarin in colorectal cancer. I'll pass over the word to you, Ulf.
Thank you so much, and it's a pleasure to be here. Why don't we go to slide number 2, which basically tells you that this is not a prospectus. Once we decided that, let's go to slide number 4. There we go. So why is Isofol a blockbuster opportunity? Well, first of all, colorectal cancer is a very common tumor. It's the second deadliest, third most common, with an estimated increase in incidence over the next 2 decades of over 60%. Arfolitixorin has the potential to be the first drug for all-comer metastatic colorectal cancer patients in almost 20 years. Arfolitixorin is expected to target the addressable market of approximately $1 billion in peak sales. Data analysis process has begun on the fully recruited pivotal phase 3 study. Isofol has already 2 strategic license agreements, one with Solasia in Japan and the other in Canada with Paladin Labs, at roughly a market value of $123 million. We have secured a robust IP protection, now worldwide, US, Japan, Europe, rest of the world to 2038, and we're working on prolonging it even further. We also have a very strong financial position, and we were listed on the Nasdaq Stockholm in the mid-cap segment in October of last year. Why don't we go to slide number 5? So basically the market, it's as I said, a very large and underserved patient population. It's the third common cancer, most common cancer, and sadly, if you have a disseminated metastatic cancer, only 10% of patients survive the 5-year limit. And the incidence is increasing. The market is also increasing fast with novel drugs coming in the second, third line, and now we in the first line. If you go to the next slide. So why would you develop and commercialize arfolitixorin? The folate-based prodrugs, leucovorin and levoleucovorin, are currently used in combination with 5-FU today and has been for roughly 40 years. They have to be converted in the cancer cells to the active metabolite before they can be effective. And the active metabolite is what's called 6-MTHF. Nearly 3/4 of patients are unable to support that conversion, resulting in poorer clinical outcomes as compared to those 1/3 who can. Isofol Medical is working to safely unlock the full strength of 5-FU with our compound arfolitixorin. We've done this by re-evaluating how an active folate can increase the efficacy and potency of 5-FU without negatively influencing the risk-benefit ratio. Arfolitixorin consists of the active substance, 6-MTHF, requiring no conversion at all, and has the potential to combine with 5-FU more effectively to inhibit the DNA synthesis, resulting in a greater likelihood of tumor shrinkage, better patient outcomes. Arfolitixorin has the potential to be the first and only immediate active folate that bolster 5-FU activity, enhancing the tumor kill effect without increasing toxicity by the chemotherapeutic agents. The goal is to make the next generation of 5-FU-based regimens as safe as those used today and clearly benefiting patients with cancer. Let's go to the next slide. Arfolitixorin as an opportunity potentiating the 5-FU efficacy. Well, we have an established mechanism of action. It's well known since 60 years back. We have the first pure form of stable form of 6R MTHF, requiring no metabolic activation. FDA and EMA granted us approval to initiate a first-line pivotal registration study in the metastatic setting of colorectal cancer as an improvement of established care based on, first of all, a very favorable safety profile and an established mechanism of action. We have preclinical and clinical documentation, and that resulted eventually in that FDA gave us the fast track designation for our R46. We have a lot of positive clinical data. PK/PD data demonstrated significantly increase of MTHF levels in cancer cells compared to standard of care. Our phase 1/2 studies showed an efficacy of close to 60%, 59%, and a disease control rate. That means you take all the responders and the stable disease together of close to 90%. And RFOX is the backbone of our registration trial. We also have a full CMC process in place. We've been working with Merck and Sai and Recipharm over 15 years to develop and manufacture the process and the know-how. And now we can say that CMC and the commercial scale manufacturing process have been completed. So this is a good step. If we go to the next slide. There have been no new treatments for what I call all-comer metastatic colorectal cancer patients in almost 20 years. And keep in mind what's going on around colorectal cancer. Breast cancer has a lot of new treatments, much more effective than what we had for 20 years. Lung cancer the same, hematological disease the same. But for colorectal cancer, basically nothing has happened. So all major national clinical guidelines recommend the use of 5-FU-based regimens in the first-line treatment of metastatic colorectal cancer, a recommendation that will stay for the foreseeable future. Most new targeted therapies are only appropriate for small fractions of the metastatic patient population, patients with identifiable mutations that are key for cancer cells to survive. For example, microsatellite instability, impaired DNA mismatch repair, or BRAF gene mutations. Those mutations have been also targeted for new drugs, but they haven't reached the first-line setting yet. There are only a few drugs currently being developed in the first-line treatment, whereas most large-stage candidates are focused on later treatment lines and directed, as an example, with the niche populations. If we go to the next slide, a little bit about this study. So we have completed a pivotal phase 3 agent study in the first-line setting of metastatic colorectal cancer. If you look on the design, it's a very conventional phase 3 study. R46 with 5-FU-oxaliplatin and bevacizumab compared to FOLFOX6, modified FOLFOX6 with Avastin. So standard of care versus our drug RFOX, only switching out leucovorin with R46. The primary objective of this study is tumor shrinkage, overall response rate, and then a key secondary objective is progression-free survival, i.e. the time it takes for the tumor that has responded to the treatment to start growing again, or the patient dies. This study has gone through a number of safety evaluations by the independent IDSMB, and we have had no flags at all. And the sample size is adequate to potentially reach or surpass the regulatory goals. If we look what we're trying to aim for, of course, the study is as directed by the regulatory agents. We need to increase the efficacy on the primary objective with at least 10%. So roughly compared to historic controls from 40 to 45 to 50 to 55%. And the progression-free survival, we need to show a statistical trend of at least 1.8 months if the standard of care is 10 months. And of course, we don't want to increase the toxicity, so preserve the risk-benefit ratio. If we go to the next slide, a little bit about the update on our study. So we have begun to do the readout and analysis of the agent study data that will lead eventually to the unblinding of the database, and we will then be able to extract the top-line data and the final results. This decision follows the discussions we had with the FDA on the censoring rules and the number of required PFS events to start the analysis process. Our database now consists of 490 patients, and we will analyze the data both with the original and also the new censoring rules in the application if the data holds up for a new drug application. Both censoring rule analysis will be included. The integrity of this of the agent study remains strong, and the team now at Isofol is very focused on doing a comprehensive analysis and looking into the data before we can communicate the top-line data. If you go to the next slide, what have we achieved? A little bit about the highlights from the first quarter. First of all, we had a close and frequent dialogue with the FDA regarding obtaining the agency approval for starting the statistical analysis based on this new updated statistical analysis plan. And thanks to the fast track designation, we were allowed to have this intensive discussion with them. We're also planning for the pre-NDA meetings that are upcoming in the second half of the year. We also have a proactive clinical preparations to ensure the proper collection and handling of the data in the analysis of the agent study data. We're also working on updating different outcome scenarios, basically guessing if the data looks this way, the data looks that way, and then how to associate that with go-to-market strategies. And this is done a bit on the basis of the information we gathered on payer and market research. And we also have had a number of productive conversations with new and potential partners. We're also strengthening our US awareness activities, so PR and communications, medical affairs, and I'll come back to that a little bit later. If we go to the next slide. So again, what have we done and what are we in the process of doing to achieve a value creative business development discussions, and also preparing for a good and successful launch as early as 2023. So we completed the recruitment of our second field member, the MSL. We have updated the medical narrative. We're in process, again, of expanding the MSL's workforce in the US. And we're preparing for ASCO and the Bio International meetings. On the market side, we updated the market and payer analysis, the demand market research, the landscaping. And in communications, we now have a US-based communications agency commissioned. And we're in process of basically looking at our communication strategy and positioning. So important things that have been happening under the first quarter. And of course, we're pursuing aggressively during the second quarter now. If we go to the next slide. So again, looking at top line and final data. So top line, keep in mind that's the first glance at the key endpoints from the clinical study. Presentation of this data will not include any detailed figures, etc. And this is because we are saving a number of the data for meetings and like ASCO and ESMO. And you're not allowed to present data that has already been presented. During the period between top line and NDA filing, additional data will be included. Current data matures. And looking at the clinical study report, the final data, it's more detailed information from the complete study databases, including then subgroup analysis that are generally planned for presentations at medical congresses and inclusion in peer-reviewed journals. So big steps in front of us. Next slide, a little bit about our strategic priorities, which hopefully paves the way for a success. First of all, we're focusing on a diligent, efficient, and high-quality approach of analyzing the data, leading hopefully to an NDA submission to the FDA if the data holds up. We're continuing to execute on our strategic partnerships in Japan and Canada, and also pursuing dialogues with additional potential partners, licensors. We're looking at strategically to evaluate the go-to-market opportunities based, as I mentioned, on different outcomes of the study. We're building also the arthrolytics awareness and value through accelerated pre-commercialization activities. And one of those is, as I mentioned, having medical scientific liaisons on the ground in the US to talk to thought leaders, key opinion leaders, and others to increase their awareness of a drug that may be coming to the market quite soon. If we look on upcoming milestones, as everyone is anticipating, we hope to get the top-line data very soon, within 4 months. And that will include the primary efficacy endpoints and also secondary endpoints. Once we have the final data and an NDA application, we're still within this year, so end of 2022. And once you submit the NDA, the FDA has a year to review it with the fast track designation. We can shave off time here. And once you get your approval from the FDA, you launch immediately thereafter. So if I may conclude in the investor highlights on slide 15. There is no branded competition in key markets where 5-FU-based regimens are dominant. That's an important variable. No branded competition, no sales force out there. Data analysis process has begun in our late stage, fully recruited phase 3 study. Isofol's phase 1/2A study demonstrated a 59% objective response rate in the Arfox regimen with no safety issues at all, the cytotoxic backbone in our pivotal study. Isofol has taken a more structured approach to protect our IP compared to other biotech companies, and we now have patents valid until 2038 in the whole world. Strategic CMC partnerships with Merck and Sai and Recipharm have been in place in 2013, and strategic licensing agreements with Solasia in Japan and Endopaladin in Canada since the last 2020. Time goes fast. Pre-commercialization activities are in process, including then the development of medical affairs and commercial launch packages. So again, Isofol is aiming for the first launch in the US as early as 2023, supported by this fast track designation. So I'll stop there and take questions. So over to you.
Great, thank you Ulf for that presentation. A few questions then. First one, you mentioned arfolitixorin is the active metabolite, doesn't require conversion. And I'm curious to hear whether the improved efficacy that you're hoping to show, whether that's driven by showing an effect in slow metabolizers, or whether you're also expecting that there is sort of a greater efficacy also in normal or fast metabolizers, if you will.
So the key thing here is when you use prodrugs, this is a general observation that prodrugs are less efficacious than the active metabolite. So in our case, we're taking out prodrugs that needs quite complicated conversions into the active metabolite. And we have demonstrated then in PK and in per-operative studies that if you compare to leucovorin, we get significantly higher amounts of MTHA intracellularly. That's one thing. The other thing is that this way we are giving arfolitixorin in the study by a fast bolus infusion of 2 to 3 minutes compared to the old way of giving it means that we peak the amount of MTHF that we get in. At the same time, we're also giving 5-FU as a fast bolus infusion over 3 to 5 minutes. So basically, we're saturating the degradation of 5-FU. At the same time, we're increasing the levels of MTHF. Those two together should boost the efficacy of 5-FU. And that should result then in that the regimen that we're using, Arfox with Avastin, or even the Arferi, which is using irinotecan instead of oxaliplatin, should increase that regimen very much. We're hoping then, of course, to achieve more than the regulators specified for us. So again, and this is why we're working with different scenarios, is that as we are blinded to the study, we don't really know what's going on there. We feel comfortable about the safety. We've had 7 looks through the DSMB without them signaling any issues. And of course, everyone hopes to have, really, really good results. But I've done, I think, 13 phase 3 studies in oncology, and the results have never been what we thought them be. So you need to be humble here until you have the data and see what you're playing with. That's basically what I can say at this stage.
Alrighty, good. And I guess on maybe a tricky question, but on this this question around updating the censoring rules, that came as a result of that unexpectedly large number of patients proceeding to another or subsequent treatment. Do you have or what can you kind of say around what could have been the cause of that, or what are the potential scenarios that you see having driven that?
So compared to other cancer forms, metastatic colorectal cancer is a slow mover, I would say. This means that a lot of patients go for very, very long, extremely long periods of chemotherapy. We're talking about patients going for 10 to months over a year with treatment. And chemotherapy is tough on the body, especially if you also are very, very sick. So a number of patients, and this happens in every day in basically wherever chemotherapy is used, patients get fatigued. And what you do then as a clinician is that you start reducing the number of drugs that you're working with. So in our case then, it's allowed in the study to take out oxaliplatin because that has cumulative toxicity. It's allowed to take out Avastin as well. What you're not allowed to take out is leucovorin, of course, which is the key drug here that we want to increase the efficacy of. There is another alternative to 5-FU leucovorin or 5-FU arfolitixorin, and that is capecitabine, an oral agent, 5-FU-like activity, with significant side effect, hand-foot syndrome, etc. But for patients who are sick, have gone through numerous chemotherapy regimens, this may be a way to limit the exposure for these patients. They don't need to come to the hospital as often. They can take an oral pill rather than coming to the hospital and getting bolus infusions. So I think that's what's happening. What we do know is that in the pool data we can look at, we have a number of patients going for a very long time. So that would be my best guess.
Alrighty, okay. Maybe just one more question then. I guess there is some degree of uncertainty or some question of how regulators would act in a scenario where you get differing results depending on whether you're using the original or the new censoring rules. And I guess maybe two parts to it then, kind of your degree of confidence in a favorable outcome in such a scenario wherein, let's say, the new ones show more favorable results and the original ones fail to do so. The first part, and then maybe a second one on whether this, whether you think this potential uncertainty then could impact how able you are to potentially strike a licensing deal prior to the FDA approval. If this uncertainty wasn't there, would there have been sort of a greater likelihood of striking such a deal prior to the FDA approval, or how does that sort of impact your thinking around funding and so on?
Yeah, let me take the last question first then. Of course, the magnitude of benefits seen in the data from the study will determine when a deal can happen. So I would anticipate that if we have stellar results, you would see the companies now that are basically hovering around us to see what the top line results are. They would be coming down like hawks onto us if the data is very good. If the data is iffy in the way that, no, sure, you met the regulatory goals, but the progression-free survival, time to events, and keep in mind, we're also looking at overall survival as a safety endpoint. And we won't have the overall survival until basically we have the NDA package done. Then I would say that if I was a potential buyer of the company, I would rather prefer to wait until I see the time to event data being matured. And so those are the scenarios I can see. If I look on this first question you asked about the censoring rules. So it was a bit surprising that the FDA were not opposed to us submitting both analysis done by the per protocol censoring rule and then to the new approved FDA approved censoring rule that is included in all new protocols that have been done since 2021. Of course, the FDA would analyze the data according to the protocol initially. Would there be a discrepancy between the old censoring rule and the new censoring rule? Common sense has to prevail. And what the FDA default answer is when you ask them is that it depends on the totality of the data that we are looking at. So you have to weigh in the clinical efficacy compared to the control arm, but also the safety and other analyses that have been done. And then it's the clinical, by the clinical reviewer in the end that makes the decision if this is something they think will benefit patients with colorectal cancer.
Okay, no, that's helpful. I think we're all out of time. Thank you very much for that presentation, Ulf.
Thanks so much and have a good meeting. Bye.
Hi again. Our next company is Guard Therapeutics, a company working on treatments for kidney injury. With us, we have here today Tobias Agervald, the CEO. Tobias, thank you very much for being here, and now the word is yours.
Thank you very much. So my name is Tobias Agervald. I'm the CEO of Guard Therapeutics, and glad to be here and present our company. And just to give you a sort of 1-minute summary of Guard Therapeutics, our projects, where we are today, this is just one introductory slide. So we're a clinical stage biotech company, and as heard, we're developing targeted therapies for patients with different types of acute kidney injuries. We recently transitioned into phase 2, so we're currently running a large global phase 2 study called the Akita study, and I will come to that. That includes 268 patients for the treatment of AKI in conjunction with open chest cardiac surgery. So open chest cardiac surgery, there are multiple reasons we're going into that space. Of course, because we have a good molecule for that specific indication, but also because there's a large unmet need and a significant commercial opportunities. We see a plus $1 billion commercial opportunity in this space with regards to peak sales. And as you also will see, there's a limited competition without any approved therapies. Also in this indication, the recruitment is projected to be relatively quick, at least in comparison to many other types of indications, which means we have data coming through relatively quickly. We have first an interim data analysis expected in the first quarter of 2023, and we have top-line results expected towards the end of next year. I will not discuss preclinical data today, but we have validated our lead asset called RMC-035, also referred to as Rosgard, in multiple different disease models. And we think this is important because it increases our confidence that also the nice preclinical results we have will translate into similar effects in humans. We also know there's a well-defined regulatory pathway for approval in cardiac surgery AKI in the US, which is a main market, and this also facilitates, of course, the design of the clinical development program. So I think overall what I want to show you is that we think we have a robust project and a clear value proposition going forward in development here. And I should also mention we're a listed company on the Nasdaq First North Growth Markets. So jumping straight into AKI, or acute kidney injury. This is a very severe disease, and as heard, there are no approved therapies in this space. And actually, AKI is exactly what it sounds like. It's an acute injury of the kidneys, which leads to a dramatic and immediate decline in the renal function or the kidney function. This is often seen in critically ill patients, so there's not one sort of patient category, but for example, in the intensive care unit, there are patients following major surgeries. We have patients that have severe infections, for example, that can develop acute kidney injuries. But overall, there's a large unmet medical need. We have about 13 million individuals per year being affected. And again, AKI is associated with very high morbidity as well as mortality. And in the severe cases, these patients, they need dialysis treatment. And many patients also progress to something called chronic kidney disease, which means that they never experience any kind of recovery, but they actually have a permanently impaired and reduced kidney function. So this is exactly the space where we're going to want to go into. And to further visualize the therapeutic opportunity in AKI, we made this graph. So on the y-axis, you see kidney function, and the x-axis, there's a time component. So just walking through this, if you look at the gray line, this exemplifies a healthy subject that goes into a stage of AKI or an acute kidney injury, but actually has a natural sort of recovery without any kind of treatment. And obviously, that's not something we would like to treat or target. By contrast, on the opposite side of this, we have this orange line where you have patients that have a severe acute kidney injury that rapidly progresses all the way down to essentially complete loss of kidney function, which means that they need dialysis treatment and or even a kidney transplantation subsequently. But we also have intermediate variants of these. For example, healthy individuals that I mentioned transition to chronic kidney disease, they partially recover, but also patients with pre-existing chronic kidney disease that have an accelerated loss of kidney function, eventually progressing more rapidly into dialysis or need for transplantation. And this is exactly the therapeutic target we have for our lead asset, RMC-035, as we'll come to. So looking specifically into cardiac surgery, we know there's about 1 million patients still undergoing open chest surgeries due to what we call a bypassed graft surgeries due to ischemic heart disease. And this is often performed in conjunction with heart wall replacement. Only in the US and EU, there's approximately 500,000 surgeries being performed each year. So this is a very big patient population, obviously. What is less known is that AKI is actually very common and severe consequence, sort of a collateral damage of the cardiac surgery, where the overall estimated AKI rate is about, well, 10 to 30%, depending on the definitions you use. But if you look in enriched patient populations, for example, individuals with a high degree of risk factors, we expect an event rate of more than 50% in these patients. So our solution to this is RMC-035. This is a first-in-class drug targeting acute kidney injuries, not only cardiac surgery, although we've chosen cardiac surgery for the primary development program. So RMC-035, it's something we've made a endogenous protein based on a protein called alpha-1 microglobulin, or A1M. But we made some certain modifications to that so it becomes druggable. We produce that recombinantly, and we also have a patent protection for that. And the brilliance of this molecule is that we actually have retained function of the native A1M protein, which is essentially by nature geared towards protection against acute kidney injuries. But we have improved properties, for example, solubility. That's important when you do drug development. So I've listed the key elements here, or the functions of RMC-035, and also importantly, all these individual functions have been very nicely attributed to different parts. To the right, you see the figure, which is a three-dimensional crude structure of the molecule, and we can pinpoint specific parts of the molecule that actually possess these specific functions. So first, we have a free radical scavenging, which means that we essentially block the surge of free radicals or toxic elements that are being produced when the kidneys undergo ischemic periods during cardiac surgery. There's also a tissue deficit of something called reductase capacity, which also counteracts the generation of free radicals. And this is also another element of the molecule that we're adding to this. The third component is heme binding, because we know in cardiac surgery there's a high degree of hemolysis, which means that the red blood cells are being distracted in the heart-lung machine, and then that leads to release of a substance called heme that goes into the kidney and is a directly kidney toxic. And this is also something we block with this molecule. And finally, during ischemic period of cardiac surgery, we know that something called the mitochondria, which produces the energy within the cells, are essentially suffering, and ultimately that leads to cell death. And this molecule, RMC-035, can also improve and stabilize the mitochondria during this surgical stress and prevent the cell death. We also think the molecule is ideal for AKI treatment. As I've shown you, we really have appropriate mechanisms that targets the major disease pathways of AKI. But equally important, we have a renal biodistribution, which means that the molecule is actually being very selectively taken up by kidney cells and proximal tubular kidney cells, which is the main site of injury. So essentially, we get the compound exactly in the cells which we want to protect. There's also short systemic half-life, so the exposure in plasma goes down very quickly. And this compound is given by IV administration, so it's suitable for in-hospital treatment. And it's a really short treatment duration. I will show you that in the phase 2 design. So essentially, we treat in the phase 2 for 3 days. So moving on to the clinical studies of RMC-035. So we so far conducted and completed 4 separate studies, ROSO-1 to ROSO-4. The first 2 studies are conducted in healthy subjects. This is our pretty standardized clinical trials where you do single ascending dose and multiple ascending dose to just evaluate the PK and the safety and tolerability of the compound in healthy subjects. The ROSO-3 study was done specifically in patients with various degrees of renal impairment, mainly to elucidate the pharmacokinetics, essentially how the molecule is being eliminated and how blood concentrations of the molecule look like in different stages of chronic kidney disease. And Rostov-4 study was the first sort of minimal POC study we did. We call this as a phase 1b study, but that was done in the target patient populations, that is, patients undergoing open chest cardiac surgery. Without going into great details, we conclude on the premise of these 4 studies that RMC-035 is safe and well tolerated. We've exposed so far 52 subjects in these 4 phase 1 studies, mostly healthy volunteers, but we also had 8 subjects with renal impairment and 8 subjects undergoing cardiac surgery. And we didn't really pick up any safety signals, and it was generally well tolerated across these study populations. We have no serious adverse events attributable to study drug. And again, the PK profile of the compound demonstrates a rapid initial elimination from plasma. And we see that the exposure, though, is inversely correlated to kidney function. That means we have to reduce the dose when we give this to patients with impaired kidney function. And this is also very important when we set the dose now in the phase 2 Akita study. Despite being a PK safety study, we also did an exploratory analysis of some biomarkers, and then very nicely, this is indicative of a reduced tubular cell injury with RMC-035 treatment, and I will just visualize that. So essentially what we did is that we collected urine biomarkers that reflects cell injury in the kidney just before the surgery and just immediately after the surgery, 4 hours after the onset of surgery. And we measured a series of different biomarkers showing the same pattern. These are 2 examples, biomarkers called LFA-PB and NGAL. They are commonly used and validated biomarkers for acute kidney injury. And as expected, if you look at the baseline, that is pre-surgery, we can't really measure anything of these biomarkers because they are injury biomarkers and there are no concrete injury before the surgery. But again, as expected, once the patients undergo surgery, there's a spike. There's a really dramatic increase in these biomarkers. You see the black here in the placebo group. There's a quite significant and strong increase in these biomarkers. But this appears to be blunted with RMC-035 treatment. As you can see, the sample size is very limited. We have 8 subjects on active. We have 4 subjects on placebo. So this is clearly underpowered to really see statistically significant differences. But nevertheless, this is a very important signal, fully consistent also what we see in the non-clinical data that we have. So I think taken together, this really provides a solid basis for us now moving forward into phase 2. So today I will present a little bit about the ongoing phase 2 study, which we also call the Akita study. We thought this was an appropriate name. It's named after a Japanese guard dog, appropriate for guard therapeutics. And we also capture the AKI acronym of acute kidney injury. So this is a phase 2 study to evaluate the efficacy and safety of RMC-035 in patients with undergoing open chest cardiac surgery and additional risk factors for developing acute kidney injury. So this is a global multi-center study conducted both in Europe and in North America. As we will come to, it's a randomized study, placebo-controlled, it's double-blind, and it's a parallel treatment design. We plan to include 268 subjects in a 1-to-1 randomization, active versus placebo. There's a short treatment period of 3 days with a post-operative follow-up, including day 30 and 90 after surgery. And this design, it's a large phase 2 study, we're aware of that. So this will be a very important design to actually inform us about the best possible outcome or the best possible design of phase 3 and also facilitate regulatory interaction when we design on an appropriate phase 3 design. And just to give you a visualization of this, we have a screening period up to 30 days before the surgery when we get informed consent of the subjects that participate in the study. And then we have essentially a treatment period for 3 days. This mostly occurs in the ICU ward or potentially once the patients are transferred from the ICU to regular ward, but still during the hospitalization period. And the treatment ends at day 3, so we give in total 5 doses, the first dose being given during surgery, and then we give repeat doses up to 48 hours after the surgery. And the primary endpoint assessment is 72 hours after surgery. So really the primary endpoint is being assessed very close to the treatment period. So there's not a long follow-up time until we can actually have results and inform about the outcome of the primary endpoint. Once the patients are discharged, we bring them back to the hospital at day 30 and also day 90, where we evaluate, most importantly, their kidney function to see, also follow the trajectory of the kidney function after surgery once being stabilized. And there are multiple different types of endpoints that one can use, and this is also very important design discussions we've had. The primary endpoint we use in the AKITA study is actually, it's a binary outcome of either the patients have AKI or they don't. And we use the definition from KDIGO, which is essentially the International Society of Nephrology, that has very strict and formal guidelines of what AKI means. And we assess the presence or absence of AKI within the first 72 hours after surgery. But of course, we can look at changes in kidney function in many different ways. So we have key secondary endpoints, which include that we look at the time-corrected area under the curve of the kidney function, including the plasma biomarkers we use to assess kidney function. We look not only at the presence of AKI, but also the severity, the duration, and the persistence of AKI, because, of course, you could have mild AKI or you could have very severe AKI, and those are completely different sort of clinical manifestations. We look specifically at the presence or incidence of dialysis treatment and a composite endpoint called MAKE, standing for Major Adverse Kidney Events. And this is a composite of either death, dialysis, or more or equal to 25% decline in kidney function as compared to the level they had prior going into surgery. We will do a blinded interim analysis when we randomized and treated 134 patients. That is exactly 50% of the anticipated recruitment. And we expect to get data from this blinded interim analysis the first quarter in 2023. And this will be conducted by an independent data monitoring committee. And in essence, the principal outcomes of this analysis will be based on pre-specified criteria. So either we continue as planned up to 268 subjects. We have the option to expand the sample size up to 348 subjects based on certain conditions. But of course, there's also an element of futility and safety analysis. So if we really see that there's no effect at all, or if there would be a safety concern, the study can be prematurely terminated. But these are the major sort of outcomes. When designing the AKITA study, I just want to recognize that we really have world-leading experts in this space. To the left, we have 3 European experts. We have Professor Alexander Sabok in Münster, who's also the lead investigator of the AKITA trial, who's an anesthesiologist. We have Professor Andreas Böning in Gießen in Germany, who's a cardiovascular and cardiothoracic surgeon. And we have Professor Claudio Ronco, who's an Italian nephrologist. And in the US, we have representation by Daniel Engelman at Bay State, Massachusetts, and who's also a cardiovascular surgeon. And we have Professor Jane Coyner in Chicago. So we really have a good representation of all the different sort of specialties that are being involved and interested in AKI and also in the clinical management of these patients. Just a couple of words on the commercial opportunity. Of course, there's different way to estimate, to assess this. In a simple way, we could say we just look at what's the overall patient population undergoing cardiac surgery in EU and the Big Five in Europe. That's about 500,000. And based on the enrichment of higher risk individuals in the AKITA trial and also in the envisioned phase 3 trial, we think that there's around 200 patients that would be eligible and would be the initial addressable patient population in a commercial setting. So depending on the estimates, of course, we have different price ranges, and that also depends, of course, depending on the outcome of your phase 3 and also the endpoint selection in phase 3, but you end up in peak sales up to $2 billion. So I think no matter how you look at this, there's really a substantial and significant commercial opportunity. Of course, before going into a big study like AKITA, we need to make sure we have a solid IP position. So we have composition of matter patents covering both RMC-035 and related variants, and that has already been obtained in all the major markets, EU, US, and Japan, which expire in 2037. So that gives us a solid patent protection, also with the possibility for patent extension. We also have a few additional medical use patents with variable expiry from 2029 to 36. And we also have priority patents in our portfolio related to the discovery platform that I won't discuss specifically today. We also get the question about competitors. So this is a very interesting space, I would say. We see emerging competitors, companies investing in this, mainly biotech, but also big pharmas now going into AKI and cardiac surgery AKI. But as said, there is no approved drugs, and I think we're advanced. There are no really companies that are in phase 3, that as far as we know. And this is just a way to kind of list these companies, the mode of actions of the compound, which are different than RMC-035, and also the different modalities or the disease pathways that we think is important. So, for example, the multi-target approach, the ischemic injury, mitochondrial binding stabilization. We have the heme binding component, the free radical component, kidney being the primary target organ for the treatment and supporting of tissue generation. And I think in essence what we see here is that we really click all these boxes, and I think that also makes sense based on the mechanisms that we have. And we have pretty high confidence that RMC-035 is definitely the first in class and could also be the first to market in this space. And as a final word, we're expanding our pipeline opportunities. We are looking actively now, not only in cardiac surgery, but this is a suitable molecule also for other types of AKI indications like kidney transplantation and sepsis. We're looking very concretely into kidney transplantation right now to potentially expand the program. And we have more assets now coming through our discovery pipeline, which is also suitable for renal diseases, but also other non-renal diseases. So rounding off, these are the key milestones we can expect during 2022 and also in the upcoming year. We expect to be able to submit applications for an IND opening and also fast-track designation in the US in the second half of this year. We have, as alluded to, the interim analysis of the AKITA study coming up with top-line results towards the end of next year. We're, as I said, actively looking into start of a clinical program in kidney transplantation, and obviously that's something we will get back to in more detail. And similarly, we have now promising preclinical data of new molecules coming through with very nice efficacy also in for example, various types of AKI models. So we expect candidate nomination and even to bring additional assets forward to make them phase 1 ready. So a lot of significant value inflection points on the way. So with that, I hope I've given you a good overview of Guard Therapeutics, where we are in the project, and I'm happy to take any questions. Thank you.
Thank you very much, Tobias, for such a nice presentation. And now we have time for a couple of questions. And my first question will be around your primary endpoints, primary endpoint on AKITA and your secondary endpoints. Because you have said that your primary endpoint is a binary outcome. Either you have or you don't have acute kidney injury. But your secondary endpoints are also very relevant. For example, severity. In a hypothetical case that you would not see a significant difference in your primary endpoint, but you see in the secondary endpoint, for example, severity, would that be enough in order to move clinical development forward? Or one doesn't make sense without the other?
So that is a very, very good point. And we call the AKITA study an exploratory phase 2 study because, as you say, in a clinical study, you have to pinpoint, you have to select one primary endpoint. And there was a lot of discussions around this when we finally ended up with the binary primary endpoint. The benefit of having that is it gives us very nice power, actually. So we're very well powered for looking at all the secondary endpoints. But you're completely right, and I think that's a point I've also tried to make earlier, that you could still fail on the primary endpoint because it's a binary outcome if you have nice effect and you can visualize a renal protective effect. So I think if we see consistency across several secondary endpoints, even if you would fail on the primary, that itself could be very relevant, clinically relevant, sufficient, and would also put us in a good position to have an informed discussion with regulators, how we would set up a pivotal program in this space.
Very, very interesting. And my final question is, after AKITA, it's still far from today, it's 2023, but what's your strategy to move forward to a potential phase 3? Is it you would like to do it alone, or you're planning to do it with a partner? Do you have any thoughts on that?
Yeah. So I think it always at this stage, you have to keep all the options open, of course. I think if you have a new, potentially first in class drug, first to market drug, with nice clinical data in a large phase 2 study, I think the most reasonable option would actually be moving forward with a partner on board. And I think if that becomes the outcome of the Akita study, I think we're in a very good position to move forward in the best possible way.
Okay, interesting. Thank you. Tobias, thank you very much for being here with us today and for your nice presentation.
Thank you.
Welcome back. The next company to present is Egetis, and we're joined by CEO Niklas Westerholm. Warm welcome. Egetis is a Swedish company, market cap around 650 million, in a pivotal trial for its lead candidate Emcitate for a rare and disabling genetic disease called MCT8 deficiency, an orphan disease. I'll hand over the word to you, Niklas.
Thank you, Adam, and a pleasure being here again this year. As Adam mentioned, my name is Niklas Westerholm. I'm the CEO of Egetis Therapeutics, for those I haven't had the privilege to meet before. What I'll cover today is an overview of the company, our strategic direction and the pipeline, and then narrow down a bit on our lead candidate, Emcitate, both from a development program perspective, but also calibrating the commercial opportunity. So, Egetis Therapeutics, as Adam mentioned, we are a small biotech company based in Sweden. We have a clear strategic direction, which is in late-stage development of treatments for rare diseases, with the ambition to commercialize the assets ourselves in Europe and US. We have 2 assets in the pipeline today that are truly late stage. We have our lead candidate, Emcitate, for the treatment of MCT8 deficiency in phase 3, and we have Aladote for the prevention of acute liver injury caused by paracetamol poisoning, also embarking on a phase 3 study during this year. Clear regulatory pathway for both candidates. We're targeting an MMA and NDA submission for Emcitate next year, and for Aladote during 2024, back end or early 2025. The commercial opportunity for both assets are very attractive, and mentioned before, we are planning to commercialize this ourselves in Europe and US, where we are now in the process of building up our commercial infrastructure and executing on pre-launch activities. That to a side, we have also, during the last couple of years, built up a very strong in-house organization in late-stage development and registration, with focus on orphan assets, and that is something we're going to continue to do over time. Furthermore, also worthwhile mentioning it is that we are building a sustainable company. We continue to look for additional assets in the orphan space that we can bring into the organization, optimize the development pathway up until approval. So that's something that's going on in parallel with our development programs. I thought I'd take an opportunity actually to take a step back and reflect, and especially on the tremendous momentum we have had over the last 4 months. So with the focus on Emcitate, we have made significant progress towards marketing application both in Europe and US. This is based on clinical data from our first clinical trial that was complemented with our publication in October last year, which is long-term data following these patients up to 6 years. That led us to further intensify the dialogue with the European agency as well as the FDA. With the EMA, we had a pre-submission meeting in December where we actually agreed that the data we have at hand would be sufficient for a submission for marketing authorization application based on existing clinical data. Then one can always ask, why don't you submit today? And the reason for it is that the number of different components in the regulatory dossier needs to be completed, and the one that is outstanding, which is ongoing, is the CMC part of the dossier. In a similar way, we had a good dialogue with the US FDA with the same position, that we have strong data already available. The FDA acknowledged that, and especially on the effects we've seen in normalization of T3 levels, and also the effects we've seen on the manifestations of chronic thyrotoxicosis could be provided as a basis for market approval. However, the US FDA requested, from a regulatory standpoint, a short 30-day placebo-controlled study in 16 patients to verify the results we've seen in these previous studies. Having said that, though, of course, with now a clear regulatory pathway that has substantially reduced the risk of Emcitate or the probability of success, we see a fast way forward. But also more interesting, of course, is the likelihood of success for Emcitate, together with a priority review voucher that we're eligible for in the US. That to a side, we also have completed the recruitment for our ongoing study, Trial 2, to establish the neurocognitive development aspects and effects of Emcitate in the disease. Also, we have more than 150 patients being treated with Emcitate today on a so-called compassionate use basis or a named patient basis in different jurisdictions around the world, more than 25 countries, which is, of course, is very important to us since it verifies the unmet medical need. It verifies the physician's interest to use Emcitate as a treatment, as well as the ability to generate data outside the clinical studies. that to aside, we also have another opportunity for Emcitate. In addition to the lead indication, we received orphan drug designation both in Europe and in US during the first quarter this year in an indication that's called resistant to thyroid hormone beta. It's a different patient population, distinctly different compared to Emcitate deficiency, and here we see further opportunity outside our lead indication. So quick glance at the pipeline. We talked a bit about Emcitate and the tremendous progress made year to date with the regulatory pathway and submission next year. We also have Aladote, and Aladote is for the prevention of acute liver injury caused by paracetamol poisoning, which leads to and can lead to liver transplant, liver injury, and ultimately death. To calibrate the prevalence here, you had actually 342 people died in the UK alone last year due to paracetamol poisoning. Paracetamol is one of the most commonly used drugs in the world, that's when many of you know, but also unfortunately one of the most commonly overdosed drugs. Today there is a treatment on the market that's called acetylcysteine or NAC. It's a good drug, it caters for the majority of the patient population. However, for the patients that arrive late into the hospital, they are at increased risk of getting a liver injury and subsequent transplant and death due to the mechanism of action in the current treatment. And here we see Aladote. We're planning to start a pivotal study at some point during the next year, during this year. So a quick glance, regulatory pathway and pipeline submissions early next year in US and Europe. We have an ongoing study that is a post-approval study to establish the effects on neurocognitive development with the treatment of Emcitate. And we have an opportunity in RTH beta for Emcitate. And Aladote is about to start its pivotal study. If we then turn our attention to Emcitate, so what is MCT8 deficiency? Well, it's a genetic disorder resulting in impaired trafficking of thyroid hormone across cellular membrane. MCT8 is one of the key transporters of thyroid hormone in the body, and here the mutation is located on X chromosome, meaning it mainly affects men. Prevalence is estimated to roughly 1 to 70,000 in a conservative setting. This actually means that it's a complicated condition. An absence of a functional transporter, MCT8 transporter, means that thyroid hormone is not able to pass the cells dependent on this transporter, namely blood-brain barrier into the central nervous system, which leads to that you have too low levels of thyroid hormone in the brain, as an example. But it also disrupts the feedback loop from the brain to the thyroid gland, asking the thyroid gland to increase production. And that then means that you have too high levels of thyroid hormone in the peripheral system of your body. So again, two more or less diseases in one. You have too high and too low thyroid hormone in different tissues around the body. This translates then into some severe clinical symptoms. So starting off with too high levels of thyroid hormone in the peripheral system have impact on the cardiovascular system, increased heart rate, tachycardia, arrhythmia, as well as being underweight. And also due to this and the stress on the cardiac system, it actually drives a median life expectancy of only 35 years. On the other hand, these patients are severely underdeveloped from a neurocognitive standpoint. They have a severe intellectual disability. And to calibrate that, only 8% of the patients can actually sit up independently. So again, two diseases in one with very, very severe conditions. Our solution to this is very simple. It's the product called Emcitate with active substance tiratricol. Tiratricol is actually a thyroid hormone analog with similar chemical structure to T3 or thyroid hormone, but with the ability to pass across the blood-brain barrier without being dependent of MCT8 transporter, which means that you restore the signals and the problems in the body, addressing the root cause. We had the first clinical study that I was referring to that has driven us towards a submission. That was completed in 2018 and the results were published in Lancet 2019. The focus here was on one side of the disease initially as a primary endpoint, i.e. the chronic thyrotoxicosis part in the peripheral systems with elevated levels of T3 as a primary endpoint. It was a fairly large study, 46 patients in 9 countries. And what we saw there was very intriguing. We saw that we had the ability to normalize T3 levels in these patients more or less across the board. Highly significant, as you can see here on the slide. And what you can't see, though, it's also fast. It actually happens within weeks. Here you see the time point from baseline until 12 months. That then translated into treatment of the clinical symptoms of thyrotoxicosis. So you see at the bottom hand of the slide that weight is improving, resting heart rate, blood pressure, etc., all improved, but also markers for liver and kidney. Furthermore, and as an exploratory endpoint, we saw an indication of positive effect in the younger children on neurocognitive development. So what we illustrate here, and with focus on the left-hand side of the slide, is measurement of neurocognitive development using a scale called GMFM. It's an established scale. It's been used as an approval in other indications like SMA. It's the definition is gross motoring functioning measure. And to calibrate it here, 100% of this scale on this scale is what the ability of your 5-year-old normally has. So as you can understand, these children are in a really poor state, and 10% roughly translates into ability to turn your head, and 20% roughly translates into the ability to sit up independently. So this is something we're further studying on the ongoing TRIAC-2 trial. What we published in October in 2021 was a follow-up study that we had followed 67 patients up to 6 years. And what we saw there was exactly the same as in the first clinical study, TRIAC Trial 1. We were able to normalize the T3 levels. It was durable up to 6 years, and again, hence subsequently actually verifies the long-term efficacy and safety of Emcitate as a treatment in these patients. And furthermore, that then led us to further engage with the European Medicines Agency, EMA, and therefore we communicated that we're submitting an MAA early next year based on existing clinical data. And worthwhile reiterating, of course, substantially reduce the risk or increases the probability of success of Emcitate getting approved. The same discussion, as I mentioned, with the US FDA. The FDA acknowledged that what we have seen in previous studies with regards to treatment effect on T3 levels, as well as the treatment effects we've seen on the manifestations of chronic thyrotoxicosis, is good and would provide us a basis for market approval. However, from a regulatory standpoint, they requested a small 30-day placebo-controlled study in 16 patients where we actually can show or replicate what we've seen in previous studies. We actually have data on 9 patients that has been taken off treatment for a short period of time, and treatment have been reinstated. And there we see that without treatment, these patients move back up to their native T3 levels very, very rapidly. And therefore, that study is ongoing with plan to recruit the first patient just after the summer, with ambition to have data towards the back end of the year, early next year, and subsequently we see a submission at the mid-2023. A few words on the ongoing TRIAC Trial 2, which is fully recruited. The focus there, as I mentioned before, is to establish the effects of early intervention on neurocognitive development using the same scale, the GMFA scale, as you saw before. And here the ambition has been to recruit around 15 to 20 young children, 0 to 30 months of age. Recruitment is, as I mentioned, completed. And here we see data then in Q1 2024, which will be very important to understand the opportunity in Emcitate as a treatment for these patients. Of course, also important in pricing and reimbursement discussions, but let me reiterate, not dependent on the result of this study for market approval. If we then turn our attention to the commercial opportunity then, and what do we see there? As I mentioned before, there's no treatment approved for MCT8 deficiency today. If you look at the prevalence, that might be somewhat a bit conservative, 1 to 70,000 males, you're looking at the estimated prevalence of around 10 to 15,000 potential patients in the Europe, US, and the rest of the world where you have access to these type of medicines. Considering the value then and the value proposition here, as I mentioned also before, that MCT8 deficiency is a detrimental condition, is a significant unmet medical need, which is of course verified by the number of patients we also have on the named patient use basis. It's a low medium life expectancy of 35 years, underweight patients, a high stress on the cardiac system, but of course suffering from significant neurocognitive disability. So we have the potential, of course, to become the first approved treatment in this area that will, of course, benefit both patients, society, and the hospital setting as such. Rather than me trying to give you a price point or estimate, we try to calibrate the opportunity by illustrating it with analogs, and analogs, the analogs we have been chosen here is similar type of ultra-rare disease, which means prevalence less than 1 to 50,000 patients, targeting again a severe disease, life-threatening, very much debilitating condition, but also have the same health gains as we perceive MCT8 will have. And there it's some quite interesting analogs you can compare to, the like of Kalydeco, Spinraza for SMA, Brineura for CLN2. All of these drugs has been launched in the more or less recent years, and as you can see here, both from a global annual treatment cost as well as the rapid market penetration, it's actually quite substantial across the board. One activity that I've spoken to Adam quite a lot about has been around disease awareness and what do we do in that space. That is one of the key legs we're actually focusing on now in the pre-commercial space. And here it's around driving disease awareness at potential prescribing physicians, which are pediatric endocrinologists and pediatric neurologists. We have established and communicated last year a homepage that's called MCT8 Deficiency. We are collaborating further with patient advocacy groups and KOLs, not just in Europe and in the US. We have also established a KOL relationship with a physician in Asia. Of course, there is a huge opportunity now when the pandemic era is more or less winding down to be present at scientific conferences. We had the privilege to be present at the first scientific conference in April this year in Glasgow. We here on the right-hand side see a picture of my chief medical officer actively engaging with the physician, trying to discuss MCT8 deficiency as a disease. And that aside, we're also focusing very much on burden of disease. So we are investigating through different means, through a so-called caregiver study that we have kicked off, understanding the burden on the caregiver, both from a time perspective, quality of life perspective, but also cost. We have initiated a vignette study with prescribing physicians and understanding really the treatment journey and the healthcare costs associated to these patients. And we have strengthened the organization. So we have actually, a couple of weeks back, announced that we have recruited a new head, 3 individual senior leaders with robust and experience in orphan drug development and commercializing orphan drugs, in a new head of global medical affairs, head of market access and pricing, and head of branding and market strategy, which will be vital to drive our 2 key initiatives forward before launch. And we will continue to build the commercial organization stepwise. So that takes us towards the end. And just to reiterate, we are planning to commercialize our 2 assets ourselves. We have initiated the commercial build-up. Of course, the driver behind this is maintain a large share of revenues, product revenues over time within the company, being able to build a sustainable organization. And there are some key success factors to that. One is, of course, the high unmet medical needs, both for MCT8 deficiency, but also paracetamol poisoning. No competing compounds in development or on the market. Centralized focus target groups, actually, of specialists, so we can actually have a lean sales force engaging with that. So ultimately, we see an organization with roughly 50 full-time equivalents at the point of launch with MCT8. So with that, I will actually stop and just having the slide here with the upcoming milestones we see in front of us, which are very exciting. So I hand over to you, Adam, for questions.
Perfect. Thank you very much, Niklas. First question, I'll pick you up on something you said right at the beginning about the potential for Getis to incorporate other orphan disease assets, some slightly new. And I'm just curious to hear whether that could, feasibly happen in the near future, or would that be something sort of beyond commercialization of MCT8? Where in time are we thinking?
And I think now I'm going to give you a somewhat diplomatic answer, because it's all relative to what's happening. And of course, I think we are starting to say we are looking already now. We have actually looked over the last 12 months. And I think the process of actually finding an asset with my background and experience, and as well as colleagues, you need to look at 50 to find one. But I think it also comes back to, of course, the financial means and what type of agreement are you looking for? Are you looking to acquire something with a large upfront payment? Are you looking to in-license something which you can maybe do at a lesser cost? Or are you actually looking at, let's say, one large pharmaceutical companies that actually have a quite large dormant portfolio where there are interesting assets, which you can, as we have done with MCT8, repurpose. Those assets you can get for a decent amount of money upfront, which is backloaded. So I think it depends on what assets we find and where we find it, but we're looking to do that in the coming period, of course, is the driver. And then, of course, the other aspect to that question is post-commercialization of MCT8. One should also think about then potentially look at other assets already on the market, complementing our existing commercial organization.
Okay, no, interesting. A question then on what you were describing about this this other indication, RTH beta. You recently got that EU orphan drug designation, giving you that market exclusivity that you need. And again, sort of in relation perhaps to the MCT8 program, when would it be feasible for you guys to kick off clinical development in that? Would it need to be beyond MCT8 approval or more temporaneous?
Yeah, and I think we need to do our homework first. So again, you're going to get it depends answer here, unfortunately. But obviously we received, as you said, which we're very pleased with, ODD both in US and Europe. And just to calibrate, RTH beta is a very different organization and distinct condition compared to MCT8 deficiency, so it's a totally different patient population. Prevalence is somewhat more common, 1 to 20,000, 1 to 40,000, around that region, with symptoms more or less translating as goiter rather than as severe symptoms as you see for MCT8 deficiency. Here, from our end, well, we need to do our homework this year, looking at how does a development program look like up until approval. Obviously, the ambition would be one short study to verify this in clinical setting. If that's the case, that could be short, it could be fairly cheap. One can consider starting that fairly early, probably not this year, but subsequent years. Pending on that, we'll communicate some further details on when to start. But it's an exciting opportunity that one could perceive as somewhat low-hanging fruit.
Okay, no, interesting. That was going to be my question, what would the clinical program potentially look like? But I think you've answered that. Maybe just one last question then. We have these pilot programs for antenatal screening of MCT8 deficiency running in select areas, I think Oregon in the US and on a regional basis in the Netherlands or national. And I'm curious to hear whether there's anything coming out of that, as far as you know, in terms of impact on diagnosis rates, or indeed if there are other such initiatives in the pipeline that you're aware of, or?
No, what we've seen from the newborn screening, and as Adam said, just to clarify that as part of the newborn screening programs in Oregon, Portland, US, MCT8 deficiency is one of the checks one will do, and the same in the Netherlands. So of course that drives both awareness actually, which one shouldn't underestimate, but of course it picks up a few children here and there. But if you think about the birth in the Netherlands, it's based on the prevalence we talk about, it's like 1 children per year, 1 to 2 children per year. So I think that's important. That's something over a period of time we of course would really want to include broader. Of course it's a heavy lift because you need to engage on a national level in Sweden, in Finland, in etc. And it takes time. However, it's also worthwhile recognizing that the test for this is cheap. It's a simple blood ELISA test, and you can do it in any lab. So it should, from that perspective, will be feasible to get on. So I think whilst we drive that in parallel, disease awareness is very, very important. Again, coming back to having the physicians to understand that if you have a small boy that is 3-4 months old, can't turn their heads, don't start to develop their motoring skill, do a simple blood test. Because I think that's where we, over a short period of time, will identify the patients more rapidly.
Okay, Niklas, we're out of time, but thank you very much for an interesting presentation and Q&A.
Pleasure being here. Thank you.
Thanks. welcome back and welcome to our fireside chat with Sobi. I am joined by Henrik Stenqvist, CFO, and Anders Ullman. Henrik has served as CFO for Sobi since 2018, having held the same role at Resfarm and Meda, and also related finance roles at AstraZeneca prior to that. And Anders joined as head of R&D and CMO at the beginning of this year. He has over 20 years of experience from the pharmaceutical industry within R&D roles at the likes of Astra, Baxter and Bayer, to name a few. A warm welcome to the both of you.
Thank you very much.
Thank you very much. Warm regards from
Great.
sunny 30 degrees hot Basel.
Great. I thought we might start with a few questions on hemophilia, RSV, and the pipeline, and then move over to some more sort of financial questions. But obviously, by all means, both of you feel free to answer or chip in however you prefer. But let's make a start then on hemophilia. We've seen some very encouraging data on BIV001 or efanesoctocog alpha, with both yourselves and Sanofi seeming very upbeat about it. And we're expecting full data this summer, likely at the ISTH conference in July, it seems, with Sanofi holding a hemophilia event at the last day of that conference. My first question is, if EFAN alpha is indeed as good as it looks, there could be little debate about what would be the best treatment for achieving near normalized clotting. But the question then becomes, where is the market going? Is it still looking for sort of near normalized clotting, or is it shifting towards convenience? So my question is sort of, what is your take on that dynamic? And specifically, I'm curious to hear how you think a drug like EFAN alpha, which in a way sort of redefines normalization, how could that sort of impact that dynamic?
Yeah, as I learned early in life, that nothing is more difficult to predict than the future. But yes, taking a little bit of a quick historical look, only a decade or two back, we had as treatment goals in hemophilia patients to try to keep their factor levels above 1% to avoid life-threatening bleedings. And with more than extended activity factors and with non-factor treatments, we can provide to at least the part of the market that can afford it, treatments that keep patients about 10%, and that is what we can establish also quite well with Hemlibra, which of course improve the average care and hemostasis level quite well about where we used to be. But still, we do see too many intracranial bleedings, too many joint problems, synovitis, surgery for joint distractions, etc. So we have not controlled the long-term course of this disease yet, even with the best treatments today. And being a biologist and a doctor, I strongly believe in the systems that Mother Nature has given us, and the more we can mimic the normal physiological control with real human factors, like we do with insulin or other replacement therapies, the better. And therefore, I guess with a once-a-week intravenous injection of BIV001, where we will see the full pharmacokinetic profile in recent scientific presentations, as you alluded to. But I think, as we already communicated from top lines, we can see that for more than half the week we are at or close to physiological control, and I think we will soon see where we end up in type of trough levels. So I hope and believe that, and I think many others will do so as well, that this may be a new step of how we think about this disease. And I discussed with some colleagues that when I started early in my career in respiratory medicine, I was part of introducing modern asthma therapy at a stage when we still had only in Sweden one asthmatic dying in acute asthma in the emergency room every day, and we had only symptomatic control with bronchodilators, etc. We went into and were able to control the long-term inflammation, and we have basically more or less eradicated mortality in asthma. And I think there is a good chance that we will be able to demonstrate over time that we can provide a new level of disease management, and I do believe that this will be a strong value proposition to both patients and prescribers.
Great. No, thank you. And on the sort of commercial opportunity for Ethan Alpha, perhaps qualitatively, what should we be thinking in terms of a target market share within the factor replacement segment, and given its sort of once weekly profile with seemingly sort of hands down best in class normalization, should we?
I assume they will be significant. We have not set any distinct expectations here, and I think we first of all we need to communicate the full and see the full data set. We need to engage with opinion leaders, etc. But from everything I can see and sense, and from preliminary discussions, I'm pretty well sure that we will see this data being received with a lot of enthusiasm, and I do think we will see impact both in terms of how we develop our share of the factor market, but also in the non-factor market, etc. I think we will be able to provide value propositions that may be significant. So I'm very optimistic that this will be a great opportunity for patients in the future, but therefore also for us.
Okay. And putting Ethan Alpha aside for a moment, then looking at Hemlibra, we've seen European quarter-over-quarter sales growth sort of flattening in recent times, and a question that's being asked is whether we're seeing sort of that pool of initial eager switchers, if you will, now beginning to deplete, and you're left with sort of a pool of incremental switchers that are somewhat less inclined or eager to switch to a non-factor replacement treatment. Do you think there is such a dynamic in play, and if so, I guess what would that imply for patient losses and gains and so in relation to factor replacement and Hemlibra?
First of all, we see the flattening, and we didn't have any black and white solid data what exactly it is, but I do think it's very surprising that with any introduction of a new therapy that you getting a transfer of the more low-hanging fruit initially, and then you have a residual of patients that are of various reasons, medical or emotional or tradition, etc., more committed to the established therapy. So I think that this is anticipated and not very surprising plateauing. But I do think the other factors that we pick up and hear a lot about is that I think we as I said, we are at a kind of 10% or corresponding to 10% factor level of hemostasis with Hemlibra, and we still have patients with symptoms, breakthrough bleedings, etc. And many of these patients need occasionally treatment with factor to top up in certain situations, etc. I think all these together, I think the fact that we have a stable base of factor patients to start with, and that we can clearly identify for now theoretical and hopefully more evidence-based advantages long term. I think it's validated a little bit of the assumptions I did in my first question.
Sure. Okay. No, that's helpful. And looking at other non-factor replacements, then other than Hemlibra, we've seen some encouraging data in recent times as well. Fitusiran showed some promising efficacy data at ASH last year, but safety concerns has forced them to lower that dose and delay that program. How are you sort of thinking about the competitive threat from other non-factor replacements in sort of mid to late stage clinical development, thinking in particular about fitusiran or TFPI inhibitors?
I mean, we assume that both treatments will eventually reach the market. How and when time will tell. And we know that it's not been uncomplicated developments, and I think but we assume that there will be a place for this. We don't anticipate anything else than that this marketplace will continue to be a highly competitive field. Having said that, I think it really remains to see what are the true benefit risk with these new type of treatments coming out. And as I said, generally, if we have chronic lack of life essential enzymes that some of us are missing, I believe that the safest and most effective way of dealing with that short term and long term is if we can replace and mimic the normal physiology, and I think this will remain a core of hemophilia treatment for at least the rest of my life, I guess, and I'm sure that we will see competition. I think we will see selected patient benefit from in certain situations from other treatments, but I have little reason to believe anything else than that factor replacement and particularly factor replacement with the kinetics of BF001 will be an important cornerstone of the treatment for a long time to come.
Okay, no, that's interesting. I was going to ask you a question on gene therapies, which are nearing the market at least, but I think we've probably largely covered that in the last question as well. So I might switch tack a little bit and move on to RSV. Astra and Sanofi reported that positive data on the Melody and Medley study on the primary endpoint, at the least in terms of reductions in medically attended RSV infections, but that miss on statistical significance as it relates to that important secondary endpoint of hospitalizations in term infants. And my question is, how significant do you think that miss on significance in hospitalizations could prove in terms of, I guess, approvals, guideline inclusions, and pricing reimbursements?
Yeah, I mean, first of all, this is not a product that we are actively driving. We have a deal and we are getting incomes from the sales, but it's of course important for us and we follow it carefully. I am personally, and also with my own respiratory background, very impressed by the efficacy level we're seeing in the study. And I think in terms of relative improvement, I think we see very clearly that the level of protection against infections and for hospitalizations, and also numerically those hospitalized going into intensive care and assisted breathing are relatively close in the same range, and I think that is a consistency that I like to see. I think this p-value for that secondary endpoint was 7% and not reaching the 5%, but there was what is occurring that there was a pre-specified cross-study analysis where these data were looked together with the phase 2b study that rendered into p-value under 1 per mil. So I doubt that there will be any big hurdle to convince payers that this is not only protecting from getting the infection, but also protecting from getting into hospital. So I'm not really worried about this data, even if of course it would have been better to have seen a significant p-value also on that secondary endpoint in the first study.
Okay, alrighty. And on sort of competing programs within the RSV space, there's a number, primarily maternal vaccines, reading out this year and the next. Most prominently, perhaps Pfizer's RSV Pref. And I'm wondering sort of how, based on what you were saying there, how significant would that program showing significance on hospitalizations prove for nirsevimab, seemingly not a major issue, if I'm understanding you correctly.
Yeah, but I do think that when we see effect on protecting from the virus infection, we will see a similar effect on protection from getting into hospital. But as I said, and I don't think anyone can question that we with the AstraZeneca product do have that protection. I think the other factor is that I think generally that getting more place in the market will probably attract more attention and increase the total number of patients treated. I think nirsevimab will continue to have a very strong position. I think it's probably tricky today to come up with maternal vaccine. We have just seen all the skepticism with vaccines in the COVID space, and for nervous women in their young women being pregnant and then being offered a vaccine if they have the option to wait and see if there is a reason to treat the baby later on. So I don't think we will see a major negative impact on the market size of nirsevimab.
Sure. Okay. Perhaps a final one then on the pipeline. We've got the Emerald phase 3 top-line data later this year for Gamifant and macrophage activation syndrome, and also the SOL-1 and 2 studies reading out. And I'm curious, Anders, as the new sort of head of R&D and CMO, what program or programs are you sort of particularly excited about in the pipeline in the short or long term?
This is a little bit like asking me which one of my children do I like most. Mm-hmm. I think the fascinating thing about getting this opportunity to come into this role late in my career and working with Sobi and the orphan specialized portfolio we have is that I have to say that I cannot pick any of our R&D program, whether they are in life cycle management or early development, that I don't feel highly motivated about. I mean, I think we are. I'm in my profession and I'm in the industry mainly for I believe we are doing something really good to mankind, and there is so many evidence of that we are doing this and that we are saving lives. So whether this is with children with HLH and are getting calls from various places weekly for requesting access to treatment, etc., or whether it's when we can show effect of Kineret on keeping patients out of intensive care for COVID, or improving the life quality and the effect of treatment for PNH patients, etc. It's a very exciting, for a drug developer, I would say a portfolio like Sobi's is highly motivating, and I'm unable to pick one over the other. I can say we are not investing in anything we don't believe in.
Great. Alrighty. Thank you very much, Anders. Now kind of perhaps moving over to your sphere, Henrik, a bit more. A first question, a theme that came up a little bit in some of the large cap pharma Q1 calls was the question of the impact of inflationary pressures. And my first question then is how exposed is Sobi to some of those rising prices on raw materials, energy, services, etc. and how able are you to offset some of those inflationary pressures if indeed you're seeing them through either raising prices or other efficiency gains?
It's obviously a good question these days, but in fact, I think we already, most of us saw some inflationary pressures already during the pandemic. Thinking about, for example, transportation costs, which we were able to absorb without really any visible impact on our gross margins. And when it comes to the more general inflationary pressure, impacting our COGS, we don't really see any short-term impact, and that is because of the agreements we have with our suppliers. In terms of the longer-term impact, we will first of all see, obviously, about to what extent inflation stabilizes, which I think we hope, everybody, and then how we can compensate it. Both through efficiencies, where we will of course try to offset through efficiencies from process improvements, and then also price increases. I mean, price increases are common practice in the US, but obviously less so in Europe. And the good thing for us is that we have very strong margins. So the cost of goods is only 23% of sales. So it really takes material cost increases to have material impacts on the margins. And I think there is even a further lag in addition to the agreements we have with suppliers, and that's the fact that once you have a cost increase, you probably don't have zero inventory. You probably have some inventory, which makes the lag even longer before it starts to hit your P&L. Okay. But it's an interesting and hot topic, of course, these days.
Sure. Alrighty, and perhaps a couple of questions on operating expenses then. This year, you've guided for margin contraction, as we all know, the SPV launch driving sort of that increase in SG&A spend throughout the year. And looking beyond 2022, then perhaps what are the main moving pieces, the puts and takes that will be impacting the SG&A spend trajectory?
Well, SG&A beyond 22, well, you actually mentioned Aspaveli will continue to be there, obviously in 23 as well, and probably reaching the full impact in 24. Another important driver will, of course, be efanesoctocog alfa, both the pre-launch and the launch to establish that one. Then we have geographical expansion, for example, Doptelet in Japan, and we have SEL-212 pre-launch and launch. So we are far from ready on the SG&A front.
Sure. Okay. And then looking at R&D, we have obviously a number of Aspaveli trials getting underway or having recently gotten underway, but also some studies nearing completion, SEL-212 and Gamifant in MAS, for instance, later this year. I'm wondering then, sort of, what are the more material R&D programs in the coming years? Is it primarily Aspaveli that we should be thinking about, and sort of that trajectory of R&D spending looking further ahead as well?
Yeah, I mean, the currently and really through 2024, we have the further indications of Aspaveli according to the development agreement with Apellis. Then obviously, again, back to efanesoctocog alfa, there will be a lot of medical activities, obviously, to establish that product and to grow that product, and also post-approval studies. Then, of course, we have all the further indication possibilities of emapalumab. But you're right. I mean, if we look at the current products and the current programs, of course, R&D would come down in the midterm. So it all depends on what other assets that we add to the pipeline going forward.
Sure. And perhaps on that note then of other products, internal or currently external coming into the pipeline, a question on you have this strong cash flow. Of course, you're reducing your net debt to EBITDA down to 1.3 times in the Q1 report. How should we think about the M&A headroom in light of what you would consider sort of a target or a reasonable leverage level?
Yeah. Of course, M&A is part of the business model because we don't have any early R&D. But strong cash flow generation is of course also part of the business model, and we think that we can comfortably lever up to 3 to 4 times EBITDA, even considering the likely higher interest rates coming our way. But that's of course not a target level. That is a temporary level that we would see right after an acquisition, and then with our cash flows, we would rapidly reduce that level, as we've seen in the past. But we have not changed our view there on our debt capacity.
Okay, no, that's helpful. And perhaps just one final question then. We're reaching time. Another question that was quite prominent during various Q1 calls was on China and the impact of the regional lockdowns. Firstly, just a simple one: how much business does Sobi have now currently from China? And then what are you seeing or are anticipating in terms of an impact from those lockdowns in China?
Well, we currently sell only Doptelet to China, to our partner in China. And the size was about 400 million SEK in 2021. And we expect that to increase notably this year. We have Gamifant for primary HLH approved, but we don't yet have reimbursement. When it comes to the lockdowns and the current situation in China, well, we don't have the commercial activity there. It is our partner Fosun, but we sold quite large volumes to China in Q1, so we don't feel that we are particularly exposed in Q2.
Gotcha. Okay, in the interest of time, we have to wrap up there, but I want to thank both you, Henrik and Anders, for taking the time, and thank you very much for all your answers. Very interesting.
Thank you. My pleasure. Thank you.
All righty. Welcome back. The next company to present is X&K Therapeutics, and we're joined by their CEO, Johan Living. X&K is a Swedish, currently unlisted NK cell therapy company in mid-stage clinical development. I'll hand the word over to you, Johan.
Thank you very much, Adam. My name is Johan Living, and I'm the CEO of X&K Therapeutics. Here is our disclaimer. Please read it carefully. X&K Therapeutics, we are one of the pioneering companies within natural killer cells globally. And at our core, we have a platform technology. That platform technology is both stable and versatile and allow us to expand and activate these natural killer cells. And expanding means growing them up in very large numbers, and activating means making them cytotoxic again. This platform technology allows us to do many things in the clinic, and we can combine our autologous NK cells with different engagers, as well as using them as immunotherapy. We come quite far. We have recently published our phase 1 clinical data, showing that we have a safe product and we have very promising efficacy. This promising efficacy made us go into a randomized phase 2 trial. We currently have a randomized phase 2 trial ongoing at the Karolinska Institute in Huddinge, where we combine our autologous NK cells with an engager. In this case, it's Sanofi's anti-CD38 antibody Sarclisa, and the target is consolidation treatment after autologous stem cell transplantation in multiple myeloma. We are currently exploring how to take these platform technology further. Just before Christmas, we signed a deal with MD Anderson Cancer Clinic in Texas, in the US, which is the leading global cancer clinic. And they wanted to test our autologous NK cells in leukemia, or AML. And we currently have preclinical research going on there. We are also now exploring our platform technology and having discussions ongoing to take it further into different solid tumors. The potential of these type of companies are big, given the price points that you can achieve, as well as the patient population that you can go after. So, natural killer cells, or NK cells. Well, they are part of the body's primary immune system, and they work by recognizing patterns on the cell surface. And on a healthy cell, that pattern is in balance. So the NK cells come, search the body, find a healthy cell with a pattern in order, then it leaves it. Search for the next cell. And when a cell gets infected by virus or transformed into cancer, as a body's part of its own defense, it changes those patterns. And the NK cells come, find that the patterns are not in order and not in balance, and then it releases its killing machinery. What I also like about working with NK cells, this is Swedish basic research at its best. These cells were discovered in the mid-1970s in the Karolinska Institute here in Sweden. When you get cancer, this system stops working. These natural killer cells, they do not recognize the cancer cells and they don't kill them. What we do, the core idea about this company, is that we found a way on how to restore the cytotoxic capacities of those NK cells. How can we restart the system? And we can also grow them up in very large numbers, allowing us to give back these immune cells as a therapy. And that's the core of what we do. We have decided to work for now with autologous NK cells, which means we're using the body's own natural killer cells that we expand and activate and give back as a therapy. We believe it has a couple of really big advantages. First of all, we do not need to immunosuppress the patients because this is your own immune system. It will not be targeted by the body's own immune system. That will lead to a couple of things. First, these cells will not be attacked, so they will stay alive for longer time, which means that we believe we're going to have a longer duration of response. We can also give repeated dosages, which means we can keep giving these natural killer cells again and again and again, also allowing for a longer response time. So let's look at the technology. The technology starts by the patient coming into the clinic, donated 1 unit of blood. That is a standard blood donation. Then the patient goes home. The blood is then shipped to us, where we take out the starting material we need and we freeze it. The freezing allows us to control the production, so it's essential for us. When we decide to start the production, that's between the ball 3 and 4 in this picture, that is when the magic happens. So in a process that takes around 20 days, we optimize the conditions for the cells, they become cytotoxic again, and we grow them up in large numbers. After around 20 days, the end product is ready and we harvest it. And the end product we then put in the freezer. Since we've been around for such a long time, we have stability data on our end product, so we know it will be okay for almost 10 years. The frozen products is then shipped back to the clinic, and the patient will come in when it's time to give the treatment. The cells will be thawed bedside and then infused. The patient will wait for a couple of hours and then go home again. And that is something that will happen a couple of times. In our ongoing clinical trials, we give 3 dosages. This is a very simple process from the patient point of view. The patient will come in, donate one blood unit, and then go home. And when it's time to get the treatment, the patient will come in again, get the dosage, wait for a while, and then go home. So it's an open care treatment. If you look a bit deeper into our technology, it's a feeder cell-free system. That is, from a regulatory point of view, very good. We don't have any other third-party cells. It's a pure autologous product. And we start from frozen material. If you look at the graph to the right, you see a cartoon of the 20-day process. It starts by us optimizing the environment for the natural killer cells. They start to become cytotoxic around day 7 to 10, and then you have a rapid expansion phase. And after around 20 days, everything is ready for harvest. What also happens on these cells is that they upregulate and downregulate certain surface markers. And the good thing is it downregulates the CD38 phenotype that normal natural killer cells have. And that allows us to combine with anti-CD38 drugs. So it's essential for the clinical development. If you look a little bit deeper into the process, the graph to the left shows how these natural killer cells kills off cancer cells, and the graph to the right shows how these natural killer cells kills off healthy tissue or healthy cells. And as you could expect, the dotted line to the bottom on both graphs, that's the day 0. That is when we take out these natural killer cells from the body. They do not kill the cancer cells. The system has stopped working, and they do not kill the normal cells either. So they literally do not kill much. After 5 days in our expansion protocol, you see a slight rise in the graph to the left. They start to become cytotoxic. And after 20 days, you see a strong cytotoxic effect towards these autologous multiple myeloma cells. What is good is that after 20 days in our expansion protocol, they still leave the healthy cells, which means that they specifically go after cells that has a change in the surface patterns, they recognize that, and they kill these cells off. So they have become cytotoxic again, but they leave the healthy tissues. Our platform is protected. We have multiple layers of protection. We have 4 patent families, 2 are approved. We also have an orphan drug designation in multiple myeloma, giving us market exclusivity, both in the US as well as in EU. And of course, on top of that, we have 3 trade secrets. And working with these type of biological treatments, even if you would know how to do it, you still have to do the entire development. So we believe we are pretty good protected. So where do we take this technology then? Well, first of all, how these natural killer cells work, they work by recognizing differences in cell patterns. And then that is a general mechanism of action. So we can use this to target a wider range of different cancer indications. And we're looking for 2 conditions. First, we would like the patient to have a relatively low tumor burden. The reason for that is that at the end, this is a numbers game. Every natural killer cell will only be able to kill off a certain number of target cells. So starting from a low tumor burden, we believe we have a very high likelihood of being successful. Secondly, there are a couple of antibodies on the market today, but also in development. And they are marking the cancer cells, and on the back end, they have a CD16 handle, allowing the body's own natural killer cells to attach to them and kill the target cells. So they are marking out the killer cells for the natural killer cells to kill off the target cells. And combining these antibodies
with a very large number of highly activated NK cells, we believe it's going to have strong synergistic effects. And that is what we're currently exploring in the phase 2 trial. So, we started in multiple myeloma, a blood cancer. We have preclinical work ongoing in AML, and as I said, we really want to find a good target in solid tumor, and we can hopefully announce that in not so far future. Let's go into multiple myeloma and look into our data. We just published our first in human phase 1 trial. It's a safety trial. 6 patients treated at Huddinge. Consolidation after autologous stem cell transplantation. That's first-line treatment in multiple myeloma, a blood cancer. 6 patients, 18 dosages. And of course, we had a big array of different efficacy markers as well that we evaluated. We see a very good safety profile, but did not see any SAE. Of course, we observed 43 AEs. All of them were of grade 1 or 2, that is mild, and the majority were unlikely to be related. If you look at the most frequently reported AEs, you see shingles, upper respiratory infection, lumbago, diarrhea, headache, and paresthesia. Most of them are nothing to worry about, with the exception of shingles. And that made us both worried and excited. Well, worried, of course, because shingles, or bältros in Swedish, is not a pleasant disease. It could easily be treated. The first 4 patients were treated with high-dose antivirals, and it declined. And the last 2 patients, it was actually prevented. So we gave high-dose antivirals as a prevention treatment, and then they did not develop shingles. So why did we get excited? Well, shingles, or herpes zoster reactivation, is chickenpox virus, so these infected cells in the dorsal root ganglia. So the cell surfaces are not in balance. The natural killer cells, the highly activated NK cells, find these unbalance in the cell pattern and start attacking them, causing the reactivation. So we saw it as a marker of efficacy. We are actually about to publish more data on the safety profile of our autologous NK cells, and we are going to publish that at the European Journal of Hematology, or European Hematology Congress in Vienna in 9th of June. So keep tuned and look forward for more data to be posted. Looking into the efficacy side, there's a biomarker called granzyme B, which is part of the killing machinery of the NK cells. And I want you to focus on the graph to the right. And this is the level of granzyme B in the bone marrow. The first measurement point is at diagnosis, and the second measurement point is 4 weeks after the last infusion. And as you can see, the level of granzyme B has gone up 4 weeks after last infusion in the bone marrow. So in the bone marrow, that is where you have the multiple myeloma cells. So this tells us the NK cells have migrated into the bone marrow, and they start its killing machinery because the level of granzyme B has gone up. And hopefully, they're killing off the multiple myeloma cells. We have an ongoing phase 2 combination trial. It's a randomized phase 2 trial, 1-to-1 randomized between an antibody, an anti-CD38 antibody Sarclisa, and a combination therapy, the anti-CD38 antibody Sarclisa and our autologous NK cells. It was started a year ago. It's currently recruiting, and approximately a month ago, we treated the first patient with our autologous NK cells. And of course, this feels really good to now have the clinical trial ongoing, actually actively treating the patient. The primary endpoint is response. And of course, we are looking to a vast array of different secondary endpoints and exploratory endpoints, and really looking forward to see the results coming out of this important study. We really would like to push our clinical development further. And in order to do so, we need to have a production for large-scale clinical trials. So almost a year ago, we took a decision to start the construction of a GMP facility in Huddinge. And that facility is today readily built. We have moved in all the equipment, and we are currently validating the rooms. Hopefully, during the fall, we will be able to submit an application to the Swedish MPA, then allowing it to go into a clinical trial production. And when that is ready, we can really have the clinical development going on at large scale. The good thing also about having this in-house is that we can learn a lot. We can learn how we can drive down the cost of goods, we can learn how to optimize, and we can really learn so we can build a commercial production facility out of this pilot. As said, we really would like to work with partners. We have an ongoing study in collaboration with Sanofi and Karolinska Institute, and we have also a preclinical study in collaboration with MD Anderson in AML patients. That preclinical study is ongoing, and during the fall we can hopefully read out the results, and if that is good, we find a good target for our autologous NK cells, we're going to move that into the clinic. And as said, we are actively looking for partners in the field of solid tumors. Of course, we need to be financed as well, and a month ago we announced that we raised another 132 million Swedish krona to continue to accelerate the growth. It's a private placement. It's led by Fleury Invest, and they placed 100 million Swedish krona. Ted Fjellman, who is the partner at Fleury Invest, joined our board of directors. What I really like about Fleury Invest is that they are also owning Northex, and Northex is the production facility in Matfors in Sundsvall, where they develop vaccines, gene therapies, and other advanced applications. And getting that knowledge tied to us, I believe, is going to be a strong competitive advantage for us, because production is essential in these types of therapies. So just a couple of things about Fleury. It's a European biotech pharma investor. It has around 20 companies in the US, UK, and Sweden. It's founded by Thomas Eldred, who is the co-founder and built Recipharm. And they are based in Stockholm and London, so I'm really happy to have them as an investor. So, where are we then? Well, we are in multiple myeloma. We have completed the first in human phase 1 trial. We have an ongoing phase 2 trial. We are gearing up for our own phase 2 monotherapy study. Once the production capacity is up and running, we're going to launch that one. And we have ongoing preclinical study, both in smoldering and elderly, 2 different indications in multiple myeloma. We broaden out the multiple myeloma into AML, another type of blood cancer, where we, in collaboration with MD Anderson, are looking into different patient categories to treat. And hopefully we can take that further into the clinic, and also allowing to expand this into solid tumor. And the production capacity that's now under construction will allow us to broadly explore these in clinical trials. So, just to sum it up, XNK Therapeutics. We are a Swedish-based biotech company. We are a pioneering company in the field of NK cell research. At our core, we have a platform technology that allows us to expand and activate the NK cells. We can use these expanded and activated NK cells as a therapy, both as a monotherapy, but in combination with different types of engagers, currently on the market or in development. We have an ongoing phase 2 trial where we combine our autologous NK cells with one of those engagers, Sanofi's anti-CD38 antibody Sarclisa. We broaden out the multiple myeloma. We have a collaboration ongoing with the MD Anderson Cancer Clinic in AML patients, and hopefully we can take that into the clinic. And we are today relatively well funded as well. So thank you very much for listening in to us.
Great, thank you very much for that presentation, Johan. We have time for a few questions. And first one I was going to ask was, we've seen some fairly encouraging data from other NK cell companies, particularly in the US. That's primarily where they're based. Most of them using allogeneic cells, many of them modified. But alongside that sort of promising efficacy data, there's also been increasingly questions about the durability, and that kind of being traced back to the persistence of those allogeneic cells in vivo. So I'm curious to hear your take on that, and I guess what the implications might be for an autologous treatment.
Let's start with the efficacy question. In January, I think, data came out from a US company where they treated an autologous NK cell in combination with an antibody. And they showed very, very good data, that combination, in lung cancer. I think the median PFS was around 9 months in their highest treated dose, and compared to 1.6 months with the antibody alone, a drastic increase, showing that these synergistic effects really exist between an autologous cell and these engagers. So encouraging data for us as well. When it comes to the durability question, well, we said it from the beginning. We fundamentally believe in the autologous part because these cells will not be cleared by your own immune system. We've seen data in the allogeneic setting where the cells live for 10, 15 days. We know from our phase 1 trial, they live a month, right? 4 weeks after last infusion. They're still alive. They might be alive much longer than that. We don't know. Of course, that's something we're going to look into. But that in combination with the ability to give multiple dosages, because we don't need to immunosuppress the patient, we really believe we can get a durable response. The allogeneic ones, since they can select an optimal donor, they might be able to get a better, shorter duration, response. Might be. We don't know. But there's a possibility for that. But they're not going to be able to sustain that. So I think at the end, we said there's going to be place for both because they are different targets, there are different cancer indications that you want to go the allogeneic route. It's obvious if you have a very aggressive treatment, you need treatment today. And there are other places where you potentially want to have a more durable response. And then we'll go the allogeneic, the autologous route. And I think we are very well positioned because most companies the last 2 years has been going the allo route, as you said. So we are one of the global leaders in autologous NK cells, and I think we're very good positioned.
Good. Alrighty. And maybe one last question then. Listening to the Novartis Q1 call, they're still fielding questions and talking about getting their production up to where it needs to be for their first cell therapy, Finwire, that they launched. It's, I guess, coming up to 5 years now. And they're still struggling with aspects of that, and they're working on sort of a 2.0 production platform. So I'm curious to hear, kind of in the time since that was launched, how much of the developments in the field are you able to sort of leverage, and what are you sort of working on in terms of reducing that time from harvest to treatment?
Well, a lot. The good thing of not being the first, right? We can learn from the CAR Ts a lot. And it goes from, the production, how to set that up, to as well the vein-to-vein process. You need to be able to track these patients all the time. And when they were launched, there were no commercial systems available. Today, well, we can just plug and play. So we can significantly reduce the risks. And now also having, NorthX Fleury coming in as, a big owner, I think is something that is going to be extremely beneficial for us because we can really de-risk that in ensuring that the production, the commercial production will be as good as possible. Also learning from the pilot facility that we are now, moving into. So I think we've done something right there.
Good. Alrighty, I think we're at time. Thank you very much for that interesting presentation.
Thank you very much.
Welcome back. The next company to present is Medivir, and we're joined by CEO Jens Lindberg. Thank you very much for taking the time to join us. Medivir is a Swedish-listed company, market cap around 450 million in sort of mid-stage clinical development for its lead asset, and also a portfolio of sort of partnered or partnerable assets, also primarily within the field of oncology. I'll pass the word over to you, Jens.
Thank you, Adam. Happy to be here and present Medivir today. If we get right into it, I won't go through this slide. Important information is in our presentation, which you will find on our website, so please have a look. As Adam pointed out, we are standing on two legs. On the one hand, we do have a unique, what we believe, first-in-class lead asset in liver cancer, which we are developing ourselves. And that's where we spend the majority of our time, sort of maybe 80% of our time. Then we do have an additional sort of portfolio of assets, which we are looking for. So we have a partnering strategy where we're looking to create value by out-licensing to others. And in today's presentation, I'll focus on our lead asset, but I'll touch on the other assets, sort of the other products at the end. So this is part of a sort of a strategic shift that sort of started back in 2014, 2015, moving from sort of the research in the virology area into the oncology area, where Medivir's two science platforms of protease inhibitors and polymerase inhibitors were sort of moved into finding targets and sort of creating benefits for patients in the area of oncology. And this is where our lead asset, which is then fostroxacitabine bralpamide, which is an in-house developed asset, which we are now in phase 1b, phase 2a. And we'll talk a little bit more about that. As mentioned, so just to give a pipeline overview, these are the assets that we have. And as mentioned, I will focus on the top one. Oh, I see the box landed a bit wrongly there, but we'll focus on the top one, which is in development for primary liver cancer as we speak. So if we go into this, and before sort of jumping into the details of why we believe that sort of Fostrox is a very exciting asset to take forward, let's just quickly look at liver cancer. Liver cancer is a relatively small market today, but it's also estimated to grow quite drastically over the next 10 years, sort of almost fivefold. And partly driven by the fact that there's a very large unmet need in liver cancer. Today, we've seen a lot of advancements recently, but still, sort of only 1/3 of patients respond to the best approved combination therapies today. So unmet need remains. And keep in mind the combination part, because we believe combinations is where the growth will come from, and that's what will be needed to drive sort of benefits for patients. And it's an important element or an important part of our strategy for Fostrox moving forward. So digging into the molecule, it's a sort of very exciting sort of innovation based on two well-known sort of parts. One, with terms of mechanism of action, if you see on the right-hand side, it's based on an active substance called troxacitabine. It's an old substance. It's been developed in liver cancer, and the studies have been run, and we know it has anti-tumor efficacy. Problem with troxacitabine and all other chemotherapies in liver cancer is that it's difficult to get the right concentration into the liver without sort of creating a lot of systemic side effects. So that's why across most tumor types, you see chemotherapy being the backbone, whereas in liver cancer, that's not the case. It's used quite seldom for that reason. And this is where Medivir's sort of knowledge with regards to research in the hepatitis C area sort of came in handy, i.e. the prodrug technology was used to get the chemotherapy sort of into the liver while minimizing side effects. So on the left-hand side of the slide, you see the so-called prodrug tail, which is then attached to the active substance. And what it does is that it takes, you can administer it orally, which is a benefit in itself, and it goes through the gastrointestinal tract and it's absorbed in the liver. So we get it local targeted to the liver, enabling then chemotherapy to be used in liver cancer. And the other part, if we sort of dig in a bit further, is that while we, on the one hand, we get the chemotherapy or the active substance into the liver, the other interesting part is that when it's absorbed in the liver, we can see that it kills tumor cells, but it spares normal liver cells, because normal liver cells don't proliferate as much and therefore are not as affected by the chemotherapy. So one, we get it to the liver, and two, we kill cancer cells in the liver, but we spare normal liver cells. I talked about combinations before and why we believe that sort of Fostrox is well positioned in the area of liver cancer from a combination point of view. And that is, if we look at the current pipeline, there are a lot of development programs in place. Most of them are centered around two areas of mechanism. One, on the left-hand side, stimulation of the immune system. You have all the PD-1, PD-L1 inhibitors, etc. On the right-hand side, the compounds that block the blood supply to the tumor, the VEGF inhibitors, the multi-kinase inhibitors. Again, roughly the same. So no matter what compounds you use, there are only so many different sort of variations of combinations you can make here, which is sort of, we believe that there is a high value in providing a third bucket. And if we then look at Fostrox and the mechanism of Fostrox in relation to the others, there is good scientific rationale for why it would be promising to combine Fostrox with either one of the other mechanisms. So on the left-hand side, we know from other tumor types, lung cancer, etc., that sort of chemotherapy plus checkpoint inhibitors do add value. And when you sort of, as a chemotherapy, you induce that DNA damage and cell death, that increases the tumor antigen presentation for potential increased immune response. So there's a rationale for why they would work together, and you would have an additive effect, why Fostrox would help the checkpoint inhibitors do their job even better. On the other side, with the TKIs, it's a little bit the reverse. The TKIs, they block the blood supply to the tumor. That lowers the oxygen, and when you lower the oxygen level in the tumor, that increases the conversion to the active metabolite of Fostrox. So in this combination, the TKIs will help Fostrox do a better job. But in any case, in either one of the combinations, there is a scientific rationale for why we believe sort of it adds value, and it does add in a third mechanism to combine in the area of liver cancer. So looking then at sort of where we are and what we've shown so far, sort of last year at ESMO, we showed the first monotherapy data. We had monotherapy data in lost the slide here. Don't know if are they still seeing it on good. The first monotherapy data at ESMO, we showed in both primary liver cancer, but we also had patients with liver metastases from colorectal cancer, etc. But what we could see is that was primarily the patients with HCC or primary liver cancer that sort of showed a response or had a stable disease. So that was sort of encouraging results in showing efficacy and also showing that the safety profile is acceptable. The plan was never to take it forward as a mono, but you need to sort of start with to show that it works as a monotherapy. The plan is to develop it as a combination, and that's where we are today. So if we look at the evolution, sort of in 22-23, we're now looking to select the dose for the combination studies and then expand those studies to get sort of the first efficacy data as well in the combination. And the study that is now ongoing, you can see on the slide here, and there are two arms. First, there are two parts. One is the dose escalation sort of part, where we decide the dose for the combinations, and the second is the expansion part. But we also have two arms. One arm with Fostrox plus Keytruda, one of the checkpoint inhibitors, and the other arm is Fostrox plus Lenvima, the leading TKI. And when we've established the dose, and as long as we have a safe and acceptable tolerability, we'll roll over into dose expansion for both combinations. We're running it in EU and Asia, and I'm highlighting that because liver cancer is more common in Asian population due to higher rates of hepatitis C and B. So it's important that we also include Asian patients in the study, and we have roughly 40-45% of centers in that study in Asia, in the Korean population. The other part to comment on from study details and objectives is that the world or the treatment algorithm in liver cancer has evolved over the last year, and combination therapy of atezolizumab and bevacizumab has become sort of a standard of care. And those patients who have gotten atezo-bev in first line and progressed are also included in the study to prepare for the future. So we are now developing sort of as a first step in primary liver cancer in combination in that sort of second line, hopefully sort of earlier line setting. As a next step, if we look forward, considering that it's a unique mechanism of action, it's a class of its own, it's the only liver-targeted medicine, we do see an opportunity for it to become a sort of a backbone combination to combine with whatever other combination you're using, as long as the data in the combination study shows promise, of course. And then third step is then to look beyond primary liver cancer. We know that colorectal cancer patients are well treated today many times, but they also, a lot of the patients have liver metastases, and they are difficult to treat. So a local liver targeted chemotherapy could potentially be a benefit for those patients as well, but that will be a step later down the line. So just sort of a couple of words on the pipeline. There are a number of assets that we have either out-licensed or we're working to find partners for. We've seen some development in the last year as well. Last, we did out-license birinapant to IGM Biosciences in San Francisco, and they are now running a combination study of birinapant plus their own IGM 8444, and they are also in dose escalation mode, and they presented some really exciting preclinical data at AACR this year, where they showed sort of really strong synergistic cytotoxicity across multiple solid tumor indications. And they are now dosing up in their dose escalation. So it's roughly the same phase as Fostrox. So exciting combination there as well in terms of moving forward with benefits for patients. But as I mentioned, our focus in terms of development is primarily on Fostrox, our sort of unique first-in-class potential treatment for liver cancer. And as I mentioned, sort of three things to take away today. It's an area of significant unmet need, and considering that we have a unique mechanism of action, we do believe we're not sort of replacing or complementing others. Two, being unique, in that it selectively targets cancer in the liver, we also bypass certain resistance mechanisms for efficacy. And finally, considering that the mechanism of action is well established, it does have a strong potential for attractive combinations across both other classes of drugs in development for liver cancer. So with that, I will stop.
Great. Thank you very much for that presentation, Jens. A few questions then.
Sure.
A first one on a potential pivotal phase 2b study.
Yeah.
And the question is whether you have a sense for what the efficacy bar would be in such a study. And I'm for an accelerated approval, I guess, specifically.
Yeah.
And the reason I asked then is pembrolizumab and ipi/nivo, when they were approved, they would have had patients who had progressed primarily on sorafenib, whereas you in the second line will see more atezo-bev progressors.
Yeah.
How should we be thinking about what the efficacy bar might be for accelerated approval? Tricky question, of course. But.
Well, it's difficult to anticipate. But I mean, today, as I mentioned, sort of, or maybe I didn't mention, but to put it this way, the Keytruda or the ipi/nivo data in second line, sort of on the back of, as you said, sorafenib or those are populations that will no longer exist because those approvals are based on a population that is no longer there. So the more relevant is to look at what are you bringing as a single agent TKI, lenvatinib, sorafenib in second line. So that will likely be the comparator, where they are showing roughly 20% overall response rate. So do we need to be better? Yes. So difficult to say, but maybe up towards those 25-30% response rate, a little bit what you're seeing in first line. I think then if you land around there, then I think we are then that would be sufficient enough for accelerated approval, considering that the unmet need is so big and there's a need for additional sort of mechanisms. That would be our anticipation today.
Okay, no, that's interesting. And then as it relates to being able to secure accelerated approval with these Pembro and Ipi/Nivo likely to be converted into full approvals, I guess. Does that, from a regulatory point of view, does that make it a lot more difficult for you to get an accelerated approval in a second-line setting? Or, I mean, there is regulatory leeway in terms of bringing something else to the table and so on.
A little bit for the reasons I mentioned. We don't think so, because of them being approved in a population that is no longer the second-line population. So on the back of Atezo-Bev, it's a whole different ballgame, and it wouldn't really make any sense if you've had a PD-1 in first line. So that would you go with the PD-1 in second line? So that you probably need something different. You're looking for different mechanisms. So do we believe that they will be approved on the back of? Yes. Do we think it changes the bar? No. And going back to the 20, we will still need to show reasonably good data, but sort of in that sort of realm of maybe 25-30 if I speculate a bit. Yeah.
Okay, no, that's helpful. And you went through the trial design of the current study, and you're sort of hedging yourself a bit with the combination with both Lenvima and Keytruda. Because of that, I mean, that's one of each from the predominant drug classes, as you were saying. Beyond those two drug classes, are there, it's a shifting landscape, but are there other sort of material pipeline programs to be aware of that could sort of change how you need to think about positioning? Are there other material ones that you're...
None that we feel today or would single out. Sort of, it's a bit too early to say. Data will show. But I think that sort of what we feel good about, sort of kind of coming back to the point, is that being quite different, being a bit unique in that sort of liver-targeted approach, chemotherapy locally, which we know it works. We sort of, when we look at the landscape, we feel that, whatever targets come out and look good, there will likely be a benefit or a rationale for combining with our mechanism, which is quite different and quite unique. So as long as we're good enough in our combinations, then I'm less worried. If we are not good enough in our combinations, then we have a bigger problem. So future, no, I only sort of, the more the merrier, the better for patients, and the more combinations we can provide as well. That would be our sort of general sort of feel today.
Okay, no, that's helpful. And then on, you just mentioned that this sort of liver specific or liver targeted mechanism. Can you just clarify sort of how does that come about, or what is it? It's not something that's specifically liver targeted, but it is to do with the uptake being primarily.
So when you attach the prodrug to the active substance and you take it in orally, it goes through the gastrointestinal tract without being absorbed, and then it's absorbed. It's sort of into the liver, and there, when you get into the liver, it's the prodrug tail is cleaved off. So in order, and then that generates a cascade of sort of go from monophosphate to all the way to triple phosphate to generate the active metabolite. So it sort of kind of goes, it's like taking a fast taxi to where it needs to go. Whereas if you give it sort of IV, then you reach a systemic effect.
Sure. Alrighty. And perhaps a final question then on cash and cash runway. You've got a runway that should see you through the current study. how much further beyond that, and how are you thinking in terms of kind of trial costs or so? How much funding do you need for a subsequent phase 2b study in terms of how large it needs to be and so on? Is it?
I think I will refrain from giving any numbers on it because again it will depend on the data in that second line in the 2A space and what will the design then need to be from a size, etc. Clearly, it will be more sizable than the one we have today. We are funded through the current phase. There is the one thing, the only thing that could be worth possibly commenting is that, well, if that data looks good, it's an open label study, and if we start to see it and we choose a dose and we see that this looks really promising, then there might be an opportunity to you might want to accelerate some of the activity for phase 2b, usually some of the CMC costs. So then there might be a question of, okay, well, would there be a benefit with some additional funding ahead of that phase 2b in order to accelerate momentum? I mean, that could be a question that comes up. But it also depends on what's the promise of the data in 1b/2a, and then when we see that, then we can make a decision on it. But clearly, if it looks good, we want to minimize the time lag between 2a and 2b.
Sure. Okay, alrighty. I think in the interest of time, then we'll wrap it up. Thank you very much for an interesting presentation and interesting Q&A.
Thank you, Alan.
Hi, our last presentation today is Orexo, a company focused on pharmaceuticals and digital therapies for patients suffering from mental illness or substance use disorders. With us, we have here Nikolaj Sørensen, Nikolaj, and the CEO, and Nikolaj, the word is yours.
Thank you very much for inviting us. So I'm going to present Orexo, and thank you for the introduction. We are a company who's probably at the most exciting time of the company's history. So many crossroads ahead of us and so many opportunities that we're watching at that we have an unparalleled situation for the company ever. The basis for that is our three different business areas. We have the US pharmaceuticals, where we are promoting our product Zubsolv for treatment of opioid use disorder. I will come back to that later. We have our pipeline, where we are now have a completely new technology platform that we're branded Amorphox, which is based on an amorphous technology, which is opening up a complete new avenues for drug delivery technologies, not only for smaller molecules, but also larger molecules like biologics, vaccines, and the similar. I'll come back to that again. And then we have a new leg for Orexo's digital therapies, which is linking very tightly into our US pharma business, where our digital therapies consist of an opioid use disorder treatment, but we've then also added on a treatment for alcohol misuse and for depression treatment. So, moving into the US pharma, our main product is Zubsolv for treatment of opioid use disorder. Zubsolv is a buprenorphine naloxone product that's been marketed since 2013 in the US. It's quite similar in the clinical profile to other brands in the market, for example, a product called Suboxone, but we have some distinct advantages of Zubsolv. We have a higher bioavailability, which makes the tablet much smaller. It also results in a faster dissolve time, a better taste, as the active ingredients normally have a worse taste, and we have the broadest dosage range of any products in the market. As said, we're commercializing this with our own organization in the US since 2013, and we're right now launching in Europe together with our partner Accord Healthcare. When we look at the area we're working at, which is opioid use disorder, this is probably in the shadow of COVID-19 the worst healthcare problem in the US. We have more than 10 million people in the US are misusing opioids. We have 4 million who are misusing so much opioids that they would classify for a treatment. And we have 1.4 million patients who are in treatment. But what is worth noting is the number of people dying from opioid disorder in the US is skyrocketing, unfortunately, during COVID. It was 80,000 people per year before COVID, and we have now surpassed 107,000 people have died in 2021 from overdoses in the US, where the predominant reason for that is the opioid called fentanyl, which is a very potent opioid, more than 50 times more potent than heroin, for example. So this is the market that we're acting in, a market which is completely underserved, where we see a lot of issues patients needing pharmaceutical treatment, but even more patients needing a behavioral treatment for how to manage their addiction and come out and live a more normal life. The product that we have been promoting since 2013 is Zubsolv, as I said. It has generated more than 4 billion Swedish krona in revenues for the company. The last 12 months, the revenues was 535 million the last 12 months from Q1. What is interesting with Zubsolv is that while we've had a period of decline following some entry of generic competitors on Suboxone in 2019, we saw that several of the insurance companies in the US were opening up for competition. That was good for some because we increased our market access in some areas of the US, but we also lost some exclusive contracts where we had been the only product that was reimbursed, for example, from the largest insurance company in the US, United Health Group. That happened in 2019, and we have kind of seen a little fading sales during the last few years, but actually now in Q1 we reported an increase, and looking at the data right now, it's quite stable in the US. What will help us moving forward is that on this market access improvements, we just added on New York State, which is the second largest payers in the US. Has a gross turnover of $180 million, and we have never been reimbursed in New York State before. And take the New York State, which under the Medicaid umbrella, our market share was about 0.5%. But if you look at the part in New York State where we have been reimbursed, which is in the commercial sector, our market share is more than 10%. So this, we think, is a fantastic growth opportunity in the year from now on. If you look at this market, so the market has been growing quite fast, and there's no doubt that the need for treatment is growing faster right now after COVID than we have ever seen before. However, the actual treatment has been flattening out during the COVID pandemic. One of the main reasons for that is lack of access to physicians. But as we see the impact from COVID-19 diminish, we also expect that we will see more patients having access to healthcare. And with that, we'll see more patients who will actually get treated, which of course will be a growth driver for Zubsolv. And also for Orexo, we have had a lot of restrictions for our field force has not been able to meet the physicians as we're used to for the last 2 years. And we now see that starting to normalize, even though we're not there yet. The other thing that will help this market and also our digital therapy for opioid use disorder is there's so much going on to address the opioid epidemic. We see a lot of activities both on a federal level, on a state level, and we see a massive money inflow coming into the sector because of these opioid litigations that are ongoing in the US right now against pharmacy chains, against pharmaceutical companies who agree to pay large damages which are earmarked to improve treatment of opioid dependence. We are one of the few companies who have a comprehensive offering in opioid use disorder. And of course, this money inflow is something that we expect will help us in the future. Moving to our headquarter and pipeline. Today, we have a pipeline of 3 revenue-generating products. We have Zubsolv, as I just talked about. We also have Abstral and Edluar, which are right now relatively small in the context of Orexo. Abstral has been a very successful product, but lost exclusivity in the US and the EU in 2019. But we're still getting some good royalties from Abstral. More exciting is our platform, which is based on Amorphox. And Amorphox right now, we deliver in a nasal spray, even though we can see a much broader applications of Amorphox. And the first project is OX124, which is for opioid use disorder, or opioid overdose. And the design criteria for OX124 was to design something that is stronger and faster acting than the rescue medications available today. Because we see that the death caused by fentanyl require a stronger reversal treatment of the opioid than what exists today. And we have data on OX124 from 2 clinical trials, which is showing that OX124 is more potent and powerful than the existing alternatives. OX125 is kind of the same product, but with a different API. So that one we have right now put a little on wait because we are finalizing OX124 during this autumn for filing in the US. On top of that, we also have OX640, which is probably the most exciting product in the company because it's something where we have an ability to become the first nasal spray in the market and where we're really addressing some patient needs. And I'll come back to that in a few slides. OX124, OX125, overdose rescue medications. It's something, as I said, the number of people overdosing have reached an all-time high right now. We are on good track to file late this year. That would mean we'll have an approval in place next year for OX124. And as I said, we have a clinically differentiated product to the market leader today. So we have a product that has shown in a head-to-head comparison with the market leader today to have a faster and more powerful effect. OX640, which I actually think is probably the most exciting opportunity in Orexo because of the size of the market. This is an epinephrine product. Today, epinephrine is used for allergic shock. So when you get an allergic shock, then you take the EpiPen and you push it based on an auto-injector into your legs, and that will help you to reverse the allergic shock. Today, that is an auto-injector. The problem with EpiPen is, first of all, it's a needle, it's an auto-injector. Secondly, it has a very poor stability, so the shelf life is very short on this. So when you need it, you can't be sure that it works. In particular, if you're taking it with you outside, because it's supposed to be stored under constant temperatures in a room temperature. So if you take it to the beach, for example, you will basically have to throw it out when you come home, because if it has been in the backpack on the beach, I'm pretty sure that the temperature would be higher than room temperature. And here we have our first clinical trial expected during the summer. We have some good expectations that will be positive because we have tested this technology with 2 other APIs with success. And if we have that, we could be the first nasal alternative to the auto-injectors. And if you look at the naloxone market, when we had nasal sprays in the naloxone market, it completely took over the market from the auto-injectors. And there's no reason why OX640 cannot do the same in the $2 billion market that we're seeing for epinephrine. And if we look at the Amorphox, what is it that makes it so unique? This is actually data that we have on Amorphox on the slide to the left. It's from naloxone, where we compare to the market-leading nasal alternative today. And as you can see, it has a much higher and superior pharmacokinetic profile. So it's more bioavailable, it's faster, and it's more powerful acting. What's even more notable is that with the stability data we have is that we basically see no degradation of the product throughout even high temperatures, high humidity, sunlight. We have a product that has shown to be stable across multiple different APIs, and we have patent protections for the next 20 years at least. And we have several more patents than the 3 that have been granted in process right now. And we have tested this across a range of different APIs. And these are just some examples where you can see basically for every API that we have here, we have no degradation of the product after 12 months under accelerated conditions. That mean high temperature and high humidity. And we have also tested Amorphox in larger molecules, and that's when you come into biologics. As everyone noticed with the Pfizer vaccine from BioNTech, it has to be stored in minus 80 degrees. But that actually is something they share with a lot of different biologics pharmaceuticals, is the issue around drug delivery. How do you deliver the drug in an effective manner, and how do you have a shelf life which is acceptable for the distribution chain? And here we think Amorphox could be the solution for that. Then coming to our digital therapies. So we entered digital therapy because we saw in the opioid use disorder space that one of the main bottlenecks to treatment was the lack of opioid or of counseling services. When we interviewed the physicians, why don't you treat more patients? We know you have a long waiting list. The answer quite often was, I don't have access to good enough counseling services. So we're basically looking for a solution where we at scale could provide counseling services to patients suffering from opioid use disorder. And that led to the development of what later became MODIA. It's a cognitive behavioral therapy that you can access through your phone, laptop, iPad, which should basically either complement or even replace the need for counseling services. If you look at the macro level at digital health, I am absolutely convinced that digital health will become one of the main growth drivers in the life science sector. And the reason for that is that we have a Western world where the gap between supply of healthcare and demand of healthcare is widening. And the only way you can solve this is to find scalable solutions where patients can get access to quality health in an efficient manner, where you don't need more hospitals, more doctors, or more nurses, which would also require a higher tax rate in most countries. So I believe that digital health is going to be an integrated part of healthcare in the future. And we are one of the front runners. There are other companies, but Orexo is definitely out there in the US working to set up this industry. And the most important infrastructure and missing piece at the moment is an effective reimbursement landscape. That's something where we have been a little too optimistic for how fast the payers in the US could start reimbursing this. But we are seeing a lot of progress right now with new legislations, new codes, reimbursement codes coming out just during the last few months. And we see a significant interest, both on a political level, but also on a payer level, to find a solution for how do we put up a reimbursement system that works. It is a digital health, and even our solutions, they are disruptive. They are different from how you used to be treated. So of course, there's also a need to change the way that patients are accessing treatment and using treatment. But here we are working effectively with pilot programs, both with the large hospital chains. We have one, Trinity Health, who have been working intimately with Orexo for the last 1.5 years to develop care programs for patients suffering from alcohol misuse and for depression. We're working with a lot of the Zubsolv customers right now to test MODIA, which is our solution for opioid use disorder. And we have, I think, right now more than 2,000 patients in different programs which are testing our different solutions. And the 3 programs we have is MODIA for opioid dependence. It's a 6-month treatment, accessible 24/7 using your phone, iPad, or similar. We have one for Vorvida, which is for alcohol misuse. That's also 6 months treatment. And we have one for depression, which is a 3-month treatment. All of them are developed together with Gaia, a German company who's been around for 20 years doing these products, and is probably the most advanced digital therapy company in the world. As said, one of the key strategic features for us right now is to create awareness and experience among key opinion leaders, among patients, among physicians, and we have different early access programs, both for Deprexis, for Vorvida, and now also for MODIA, where we're giving access for patients at a very discounted price, or even some of them for free, simply to get feedback, how does it work, and also for the physicians to be comfortable of using this in a real treatment situation. And we are making a lot of progress, in particular in MODIA, where we have all of our networks from Zubsolv, where we're seeing every week we have somewhere around 40 to 50 new patients testing this with new physicians. And the acceptance rate is exceptional, the feedback that we're receiving right now. On the financial sector, so Orexo, as I said, we have our group revenue is 592 million the last 12 months. The majority of that is from Zubsolv. Zubsolv is a fantastic profit contributor with nearly 300 million in EBIT from the US pharma business for the last 12 months. Our EBITDA is negative, and the reason for that is that we have invested a lot in building the infrastructure in Orexo for digital therapy. We have tested different marketing concepts for digital therapy, but even more important is our investments in R&D. So investment in our new AmorphOX platform, 3 different projects, OX124, OX125, and OX640. And building up the manufacturing, we have basically commercial manufacturing ready for all of these products. So we're invested in that during the last year, and we're invested in a trial of 400 patients for MODIA, and that is leading us to the negative EBITDA. If I take away these one-time non-recurring expenses, the company is actually profitable, and we saw that, for example, in the first quarter here, we had a profitable Q1 on EBITDA level. I have to say, to full transparency, that one of the things that we're also spending some money on is that we have a patent litigation for Zubsolv. It has this 30-month stay. That is what you have when you have a new patent litigation, gets 30-month stay. It's going out in February next year. So we're expecting to have the trial late this year, could even be early next year, but it could very well be late this year. And we have, in 2020, we received a subpoena where we should hand over information about our marketing practice for Zubsolv to the US authorities. Nothing has really happened on the subpoena during the last 2 years, but we haven't had any outcome where they have to set whether we have done something wrong, but they haven't said whether everything looks good either. So these 2 legal risks exist. On the Zubsolv patent litigation, I will say we have been in similar patent litigations previously where we have prevailed on the federal circuit. So our patents have been validated on a federal circuit level, which makes it a very strong case for Orexo. So why should you look at Orexo? Why should you be interested in investing in our company? First of all, we have a very strong foundation. We have a profitable US pharma business where we have an EBIT margin, which is north of 50%. We have an organization in place in the US. We have the knowledge about what it takes to commercialize products. I will challenge anyone to look at products similar to Zubsolv to see if they can find anyone who have reached the same kind of market access as we have done. So we have a very, very strong market access team in the US. We have a pharmaceutical pipeline, which this new, very unique technology platform called AmorphOX could open up avenues into biologics, where we're already now talking with different companies to do feasibility studies on their biological APIs. It opens up for new product pipeline opportunities within the Orexo family. And we already have 2 products, OX124 and OX125 and OX640, in pipeline, where we have shown for the 2 first of them that they work in a clinical setting, and we're expecting to get results for OX640 during the summer on our epinephrine product. And then finally, we have digital therapies, a completely new space where Orexo is one of the first movers. We're building that on our networks from Zubsolv and opioid use disorder. So we move into a space close to where we work with our pharmaceuticals, and it provides us with a comprehensive offering, in particular in opioid use disorder, where the best treatment you can do today, everyone agrees, is what they call MAT. It's based on buprenorphine combined with psychological counseling. Today we have buprenorphine in Zubsolv, we have psychological counseling in MODIA, which create a complete offering to our customers in the US. So quite exciting situation for Orexo right now, and something to look forward during the next year. Thank you.
Thank you very much, Nikolai, for such a good presentation. And we have time now for a few questions. I would like to start asking you about digital therapeutics, the space. There has been an increasing amount of interest in this space. I mean, there has been like partnerships with big pharma companies. So it's obviously growing, the space. What do you think that it's going to be in the future? What will define? What is the quality stamp that a digital therapy has to have in the future in order to be prescribed, and not a competitor, for example?
I think there's one thing that is really missing right now, and that's an effective distribution and reimbursement system, which is aligned across all solutions. Because when we talk to insurance companies, they tell us we like the thinking, we can understand the value that this could bring, but we don't want to develop a unique solution for one company. So we want to see that there's a solution we can apply on all companies. And here we have a situation where different companies have been competing for different solutions, and there's no, universal solution for reimbursement. And I think that is right now the number one bottleneck. And if I listen to my American colleagues in other companies, it's basically the same story all of them talk about. We need to find ways to get this reimbursement system to work, because that is the key to really get paid for what people expect, and that is that we also show clinical evidence that it works. But if you're going to finance a clinical trial, you need to find a way to get it paid. So it's not only the technology development, really the big cost is in the clinical research. And we need to find a way where people want to pay, and the ones to pay it would be the insurance companies. And that kind of infrastructure is not there. So I like to say we have a shiny new train that everyone looking at the train and get to a test ride think this is working well. But for a train really to go anywhere, you need to have some train racks that the train can go on. And right now we're working intensively to put out these racks to ensure we have the infrastructure to secure reimbursement.
Yeah, and as you said, now in the US, this is advancing, and these reimbursement paths are getting more established and more established. And how does the situation look in Europe, and how do you see this?
So in Europe, it's very country dependent. So you have the most advanced country is Germany, who established a universal reimbursement system called Detta last year, where actually our partner Gaia had out of the first 5 reimbursed products, 4 of them were from Gaia. And there we're seeing that it starts to take off in some categories. So Germany has come a long way. We know that France is looking at a similar system, and so is Spain. So if I look at the 3 most advanced countries in Europe, it's actually Germany, France, and Spain. When you come to countries like Sweden or even the UK, it's much more regionalized. You need to talk to the healthcare providers to convince them to use your product, and they need to find a way to reimburse it through their existing channels. So we don't have this national reimbursement system anywhere else.
Okay, interesting. And my follow-up question is around Modia and Zubsolv. What do you think that one can influence the other one in the future? Do you see possible synergies that the launch of Modia could impact the sales of Zubsolv, or the other way around?
I mean, today all Modia is promoted in parallel with Zubsolv. So when you look at our cost for Orexo, will highlight that a lot of the selling expense for Modia or for digital therapy, actually more than half of it is from allocated expenses from our Zubsolv field force. So the Zubsolv sales reps are working both with Zubsolv and with Modia. And what we can see is that when they have meetings with Modia, they tend to have much longer meetings with the physicians. And for those clinics where they use Modia, we have much more frequent interaction. And of course, in all of these interactions, we also promote Zubsolv. So this could help us opening the door for more physicians where we haven't been with Zubsolv before and get more quality time. Over time, I think there are opportunities to make different kind of bundle agreements with larger payers in the US, with states or regions, and that could open up completely new avenues. And here in particular, I think Modia is the differentiating factor, because there are a lot of other buprenorphine naloxone products out there. We have seen a market which has become very dominated by generics with a similar clinical profile as Zubsolv, even though we don't have a generic on Zubsolv. But there are other products with a similar profile. Okay. And therefore, Modia is a very differentiating factor.
Okay, interesting. And my last question is, what can investors look for in terms of milestones in the upcoming 12 months?
I think there are quite a few milestones. In digital therapies, we have talked a lot about one of our partners, Trinity Health, which has taken a long time, but we do expect the first patients to come in now in Q2. That's our guidance, and I see no reason to change that. We have other discussions with both payers in the US for reimbursement and other caregivers. So I think any of these partnerships is a proof of principle that it would work. Then it will take time to implement before you can see real revenue growth. But to really solve this reimbursement issue is something that I think is a key. And with these partnerships, come with a solution on the reimbursement system also. And Trinity Health, that's really where we've spent our time, is to solve the reimbursement issue. So I think that's where I would keep one eye up. The other one is on our pharma pipeline, where we have this OX640 clinical trial in the summer. And if that one shows what we hope, that we have high bioavailability with OX640, we have a good position to become one of the first, if not the first, nasal alternative to auto-injectors. And again, looking at other areas like naloxone, where you have nasal sprays introduced where there have been auto-injectors before, the nasal sprays become the dominant choice. And here in Europe, there was one ahead of us, but they just had to withdraw their application. And some of the issues that were identified with that product called Neffy is actually not present in our product. It was around some of the excipients in the product that EMA were concerned about, but those excipients are not present in our product, which actually put us now as the front runner in Europe, as the only product who can actually deliver on EMA's expectations. So,
thank you very much, Nikolai,
thank you very much
being here today and for the next presentation.
Thank you for inviting us.
And with this, we finish for today, but we will be back tomorrow morning at 8. Thank you very much.